second day of International Conference on Sustainable Technologies for VIKSIT Bharat 2025. It's a pleasure to have you join us online for today's technical session 4. Today's session will be chaired by Dr. Rakesh Bhutiani sir, who is Associate Professor in the Department of Zoology and Environmental Science at Gurukul Kangri Vishwavidyalaya, Haridwar, Uttarakhand. We are also having myself, Dr. Nidhi Shukla, Assistant Professor, who is going to co-chair this program. This session will focus on various aspects of sustainable technologies, environmental science, and the challenges we face in ensuring a greener and more sustainable future. We will begin with two insightful plenary lectures, and then we will move to our contributory papers and the poster presentations that will showcase the cutting edge research and innovation in this field. We will now proceed with our first plenary lecture. At this moment, I would like to invite Dr. Rakesh Bhutiani sir for his plenary lecture. And before starting his lecture, I want to give a brief introduction about Rakesh Bhutiani sir, who is Associate Professor at the Department of Zoology and Environmental Science, Gurukul Kangri, deemed to be University Haridwar, Uttarakhand. With what 20 years of teaching and research experience, 23 years of having research experience, he has been a guiding light in the fields of limnology, ecological modeling, aquatic biology, environmental microbiology, and pollution control. He holds a PhD in environmental science along with dual net qualification from both UGC and ICAR. Dr. Bhutiani has an illustrious academic profile, having published over 120 research papers. He secured 2 patents and authored 22 books. He has supervised 7 PhD scholars and he is currently guiding 6 more. He is an executive editor of Environment Conservation Journal, review editor for Frontiers in Ecology and Evolution, and also served on the editorial board of multiple national and international journals, including the Biotechnology International Journal. Frequent contributor to the global academic community, he has delivered 30 invited talks, attending 21 conferences, who has undertaken 3 international visits, and he has been honored with 21 awards for his contribution to science and sustainability. So thank you, Dr. Bhutiani sir, for joining us. Now we are going to move with your plenary lecture. Most welcome.
Thank you, Dr. Nidhi, for a very nice introduction. First of all, I would like to thank the organizing team of MIT University for organizing this wonderful conference on sustainable technologies for VIKSIT Bharat. And by the core of my heart, I would like to thank all the organizers for inviting me and interact with the different persons of different universities to present my views regarding the environmental issues. Especially, I thanks the head of the department, Dr. Kuldeep Dwivedi, and Dr. Swapnil Rai for inviting me on this talk. I'm sharing my presentation and we'll start in a moment.
Yes. Sir, your camera is off, I think.
Actually, if I switch on the camera, there will be a disturbance in presentation. I will try to open the camera also.
Okay, no issue.
Because I had just faced it yesterday also. I was in a lecture, so they said me to turn off your camera.
Maybe some network issue.
Yeah. I will switch it on in between also.
Okay.
Is it lecture is visible to all of you?
Yes, sir. It is visible.
Yes. Please continue, sir.
Okay, just a minute. I first switch on my camera. Is it visible now?
Yes, sir. You are visible. You are visible, sir.
Okay. Okay. So we are going to present today's lecture on water management and conservation, that is on sustaining our most precious resource. Okay. Today's presentation will focus mainly on one of the most pressing environmental and developmental issues on water management and conservation. We all know that water is a vital natural resource essential for life, economic development, and environmental sustainability. And with increasing population, urbanization, and climatic change, the demand for water is growing while its availability is become more uncertain. You have made me the host. I think some people are trying to, should I admit, or you people are going to admit them?
We will admit. You just
Okay. So
we
I'm getting the message.
We are admitting, sir.
No, sir. We will immediately.
Sure. So we all know that water is a finite and irrepressible resource, and how we manage it today will define our collective future. It will define our collective future. We all know that. And first of all, I want to clear to all the audience that though it seems a very simple topic, and I will go to describe it in a very simple and a easy manner, the day-to-day routine things we can do to conserve water. So you can think, first of all, I want to aware you all that why there is a need of this simplicity, simple things that we are reminding you day by day, that is for conserving water only. Because a simple thing is that bund bund se sagar banta hai. So what I can say that if we manage it today, it will define our future. So we have to conserve the water. Now the present lecture has been, I have split it into two parts. That includes the water management, and second part consists of the conservation issues. The water management part involves the different planning, development, distributions, and optimal use of water resource, ensuring that water is available for various needs without compromising the future supply. The second part involves the conservation techniques of water that mainly focus on using the water efficiently by we people, minimizing wastes and protecting water quality through different sustainable practices. Now, if we move away from that, these approaches, that is management and conservation, these approaches are very crucial for securing water for agriculture use, industrial use, as well as for domestic use, and maintaining the ecological balance of the environment. In nutshell, if we say that about the lecture, what we can discuss in next 20 to 30 minutes in this lecture, we can say that with this session, we will explore the current water crisis, traditional wisdoms, modern technologies, and strategies for sustainable water use, especially in the context of India's development goals, or we can say that for the VIKSIT BHARAT 2047. Moving towards the next slide. Okay, this is, we all know about the position of water on Earth. Then we all know that water is an essential for all life forms, and it is the backbone of agriculture, industry, and public health. Yet, despite being called the blue planet, less than 2.5% of Earth water is fresh water. We all know that. I don't need to explain all these things. That is understood by this figure also, that only a fraction of that is accessible, that fresh water 2.5, and only out of that 2.5%, only a fraction is available. And India holds around 18% of the world population, but only 4% of the water resource are present in there. So it's a very important thing. So for further sustainable water management is thus crucial. By seeing all these data, sustainable water management is crucial not just for the development, but for our survival also, for survival of every human being, for survival of any all living organisms on this planet. Now moving towards the next slide. what I had said, that first we can discuss about the current water crisis in India. What is the current water crisis in India? If we talk about the current scenario, India is already facing a severe water crisis. Groundwater levels are depleting at an alarming rate. Rivers are increasingly polluted. Now I can let you know all please that not only in India, in around whole world, as per my knowledge, not even a single river is clean today. All rivers are polluted. Some are to some extent, some are to extreme extent, depending upon that. So no river is, we can say that it's a clear river, or the water is pure. We can't say that. So many rivers are increasingly day by day it is polluting on at a very alarming rate. Many urban areas are struggles with water, also with water challenges. We can see in newspapers, in media today, that there is a water scarcity and water shortages, water problems are there in different states of India. And if we say that, if we go to the statistics of the water in India, a report of Niti Ayog says that 21 major Indian cities could run out of groundwater by 2030. That is the report of Niti Ayog. This crisis stems from population growth, over exploitation of water resources, poor management, poor governance, and various climatic variabilities also. These are the basic things that we have to take care of. Now, what are the different management techniques? What are the different key concepts in the water management? These are the different things. I will not go into the details of all these things, but a little things I can say about that. Now, if we talk about the water management, it involves both water conserving existing water resources and optimizing their use. It includes both of the things. And to fulfill the conditions of water management, key strategies, what are the key concepts or key strategies? That includes watershed management, which generally focuses on the holistic development of catchment areas. Second one is the rainwater harvesting, in which we can collect the rainwater for reuse. Third one is grey water recycling, in which water from sinks and showers can be recycled. And the last one is the efficient irrigation, that is sprinkler or drip irrigation techniques that we are using. These are the basic technologies that are available to conserve or manage the water. First one I have already described. I will move these slides quickly. And one thing I want to add there, if we really follow these management strategies together, all these tools, including watershed management, rooftop water harvesting, grey water recycling, and all these things forms a comprehensive approach to the sustainability of the water. That includes. Now come to the rainwater harvesting. As already I have described, it is the principle of collecting and using precipitation from a catchment surface. It is a very simple and an effective technique. It is it involves collecting of rainwater from the rooftop surfaces or natural catchments and storing it into the better to recharge the groundwater. Now an example, I will give you an example, a case study of Chennai here, the state of from the Chennai, that states like Tamil Nadu have made rainwater harvesting mandatory for buildings. Now, anybody who is constructing their house in Chennai, that is mandatory for their roof water harvesting is mandatory for them. So that results in significant increase in the groundwater levels in that particular areas. It is a low-cost technique, sustainable and adaptable to both urban and rural management. So that type of examples we can take away so that groundwater and all these water resources can be conserved. Now, moving towards irrigation techniques. So different type of technologies, different types of irrigation, that is drip irrigations is there. 30 to 70% of water saving is there from drip irrigations, is from sprinkler system for broader fields. We use sprinkler systems like showers. We use, many of you have already seen that in agricultural field, that they are using the sprinkler field so that we can save water. It also saves, reduces runoff and from evaporation. And third technique is wastewater recycling and reuse. that is gray for flushing, gardening, municipal waste treated for industrial use. These all water can be reused and recycled for industrial or agricultural purposes also. Now, moving towards the next slide about the conservation of water. So we can take example from our traditional water conservation systems. That is very important. The traditional system that we have, forget about that particular our Indian traditional system. If we focus on our traditional systems, we can get many things to conserve our environment. If we see that long before the modern science, India has developed ingenious and sustainable conservation techniques. Like structures like step wells, tankas, johars, jabo systems that are found in Gujarat, Haryana, and Nagaland state. So they reflect our ancestors' deep understandings on hydrology and ecology. These systems not only harvested rainwater effectively, but also fostered community management and local ownerships, such principles that are relevant even today also. So if we go deep inside the traditional water conservation systems, we found that India has a rich heritage of traditional water conservation systems that reflects indigenous wisdoms and sustainable practices also. Adapted to the local climates and geographies, these systems were community driven. All these systems were community driven, eco-friendly, and highly effective in managing water sensitivity at that particular time. And these methods, these means that particular traditional methods not only conserve water but also supports agricultural, recharge groundwater, and fostered social cohesion and around resource managements. So there is an urgent need to revive and integrate these systems along with modern techniques. If we combine modern techniques with these traditional systems, that will give far much better results that was given in the traditional system. So these can manage today's water challenges also. We have to go to the traditional systems. In many things, we are now adapting our traditional systems. So for the conservation of water, we also have to follow our traditional system. Now, role of community participation in water conservation. The community participation is very important in conserving the water resource. Sustainable water management cannot succeed without public involvement. Remember that point. That cannot be succeeded without the public involvement. Community-led initiatives, NGOs, panchayats, RWAs, residential welfare associations, school awareness campaigns, that our government is emphasizing all these things in today's. So all these things we have to participate. Far better the community participation, better will be the results. Besides all this community participations, the government campaigns like Jal Shakti Abhiyan are also working to mobilize communities and water conservation that I have already told you in my message. Remember one thing, when local people take ownership, this is a picture that is showing when local people take ownership of water resource. I will let you know with an example in another slide, that when local people take the ownership of these water resources, the results are often more sustainable and impactful, because only government cannot do that such thing. Involvement of public community is very important, and they will give much better results. So community participations plays a vital role in the success of water conservation efforts. When local communities are actively involved in planning, they should involve in planning, implementing, maintaining all these things, water projects. If community involvement will be there, the outcomes will be more sustainable and impactful. People can contribute through, they can contribute with their own knowledge, they can contribute through traditional knowledge, they can contribute through their labor, they can contribute through resource sharing, they can contribute through collective decision making, because different peoples give different types of ideas to conserve that particular water body. If only one person will take, he or she can not be able to revive that particular water body or any thing from that. So community led initiatives like revival of ponds, constructions of check dams, rainwater harvesting, and watershed managements have proven effective all around the India. Therefore, these efforts not only enhance water availability but also promotes a sense of ownership, responsibility, and along with the team stewards of water resources also. Now, these are the slides. I can move 2 or 3 slides are there, short slides are there. These are the different policies, that is Jal Jeevan Mission, Atal Bhujal Yojana, Namami Gange, that our government is running. Besides this, community participation is different. If community participation is also involved in these policies of government, we can achieve our targets to clean water bodies and resources. Now, what I was talking about the public or the community participation. Now, this is an example, a case study of Coimbatore Lake restoration. The lake of Coimbatore has degraded due to encroachment and pollution. One side, the left side. Before you are seeing that picture, particular picture. Before that was the condition of the Coimbatore Lake. However, through a collaborative efforts involving the municipal corporations, citizen groups, and corporate CSR also fundings, because funding is very much necessary for all these types of activities. They make it clear. On the right side of the image, you can see today the condition of that particular Coimbatore Lake is this. You can see the difference between the two pictures. Restored, that means the lake has been restored by public efforts, and today they serve as thriving ecosystems, groundwater recharge zone, and recreational spaces also. So this can show, this case of Coimbatore can show that how multi-state collaborations can revive dying water bodies. This is a live example. I am showing it to you in this picture. Now, we have already talked about different management studies, strategies, conservation. How can we conserve? Now, what is the role of technology in conservation of water body? We can, as already I have said, that we can merge technology along with our traditional method. It will give more better results. So if we consider modern technology, it can revolutionize whole water management. IoT sensors, smart irrigation system monitors, GIS for watershed mapping, AI in water demand forecasting. All these technologies can help in maintaining, conserving the water bodies. Mobile apps, even people are creating the different mobile apps that helps the farmers and citizens to report water issues and assess real-time information. Also, when combined, when all these technologies, traditional systems, when these are all combined with the policies and community actions, technology becomes a very powerful for change, for any kind of change. Now, what are the different challenges ahead that we have to face? Besides, if we go forward, there are various challenges in maintaining, in management, in conservation of all these resources. So whatever efforts we can make, many challenges still remains. For example, climate change. We don't have control on climatic change that is altering the rainfall pattern. The climatic change that is altering the rainfall pattern. So we don't have any control on that particular field. That increases the water related disasters like drought, floods, and etc. Rapid urbanizations put immense pressure on existing water infrastructures that we don't have any control. Water pricing remains politically sensitive, often leading to overuse and wastage of water. So we have to overcome all these challenges. These are the challenges we are working on that particular field, so that we can conserve our water resources for our future generation also. Now, this is the basic things that I have that comes to our mind. What can I do? I have written here, what can you do? The so simple thing is that what can I do? People generally thinks मेरे करने से क्या होगा, मैं अकेला क्या कर सकता हूँ. Remember that line I have initially said in my lecture, बूंद बूंद से सागर बनता है. हर आदमी को अगर एक साथ मिलके काम करेगा तो दैट विल गिव अ बेटर रिजल्ट. सो एवरी इंडिविजुअल कैन कंट्रीब्यूट टू वाटर कंजर्वेशन इन देयर ओन वेज. अपने तरीके से वो जो है हेल्प कर सकता है. दे कैन फिक्स लीकेज टैप्स, यूज सेविंग वाटर्स, turn off the tap while brushing, fix leaky faucets, run dishwasher only when full. तो ये छोटी छोटी चीजें हैं that we can't जिसका हम ध्यान नहीं देते हैं अपने डे टू डे लाइफ में. इवन आफ्टर दैट वी कैन सपोर्ट लोकल इनिशिएटिव्स. अगर किसी ने एक इनिशिएटिव स्टार्ट किया आपके एरिया में, यू कैन support that initiative to conserve water. That will also count into the contribution of you. Don't discourage that particular types of initiative. You can increase the moral and morale so that they can help in conserving all these types of water resources. So that was the thing. Now and the last I will show you a 2 or 3 minute video is there. You can just read the paragraphs. What you just read it out and see what will happen in 2070 if we do not conserve the water in today's life. So that was the presentation. I want to show it to all of you, that if we do not conserve water, our future generation, our children will have to pay a very high price. So we have to conserve water today so that our future generation can also enjoy all these resources. Water, we know that I generally used to say that water is the very easiest and the cheapest resource available on the earth. And that is the reason we are depleting it at a very fast rate, because we cannot value the easy and the cheap resource that are available. We can't value all these things. So, and last, to conclude the session, I just want to say that water is life. Water conservation, if we go towards the water conservation, is not a choice, it's a responsibility. Also, we can say, I want to tell you know that water is not just a resource, it's a lifeline for all of us, or for all our living organisms. It's a lifeline. With India aiming to holistic development under the VIKSIT Bharat vision, water is shared resource that requires collective responsibility, sustainable water management and conservation. Just not only these are the technical challenges, but also social and environmental imperatives also we have there. Now, there is a very good quote of W.H. Auden, that thousands have lived without love, not one without water. That's a very important thing, and he said in a single line. So conserve water, help in contributing to conserve water, so that our future generation can also enjoy all these things. Thank you very much. Thank you for your time and attention, and let's work together to protect and conserve water for our future generations. Thank you very much.
Thank you very much, sir. Thank you for your insightful lecture. Thank you once again, sir, for sharing your knowledge with us today. Thanks a lot. Now,
thank you, sir, for the wonderful lecture, and I welcome you. Hello, sir. Can you hear me?
Yeah, sure, sir.
Okay, sir. Thank you for the wonderful lecture. I think the participants might have some queries. If the participants are having some queries, they can put it on the chat box, because you don't get such eminent people opportunity to interact. So you can clarify your queries and interact for some guidance with Dr. Bhudiani, sir. Any participant has got any type of query regarding the lecture that he has just delivered?
You can raise your hand. Participant, you can raise your hand if you have.
Raise your hand. You can just type it in the chat box, or you just I think they can switch on their mic and they can do the interaction.
Yeah.
Participants, you can send in the chat box as well if you have any question.
Yeah, I have opened the chat box, but no queries there.
These are the drawbacks of online sessions, sir.
Yeah, exactly.
It's always good to have the speakers live interaction, but due to certain constraints on the part of the organizers, as well as I will say on the part of the speakers also, online has become a medium, although it does solve some issues, but the fruits of interaction that we get in a offline interaction, that seems to miss in the online session.
Exactly what, if I correlate your point with my lecture, I am just saying that we have to move towards our traditional system only.
Yeah.
Then only we can get better results.
Yes. I agree with you, sir.
In each aspects of life, not only in conservation of water, not only in technology is good, but we can't depend totally upon technology only.
No, sir. Next time, sir, we will have you in person here in our
Sure.
university. And this time also we tried to have this whole conference in the offline mode, but the participants particularly are still reluctant to make the efforts to travel and come to attend the conferences and all that. And because of that only we had to make it in a hybrid mode. Otherwise, yesterday we had in addition to the inaugural session, we had one session of
Offline.
4 speakers were there in the offline mode itself.
Okay.
And then next time, I think we will have to remove the option of online altogether.
Exactly. Yeah.
Right. Anyway, sir, thanks a lot. And it was nice lecture from you, a lot of information. And if the participants would have listened to it carefully, they will definitely derive benefit of the contents of the lecture that you have delivered. Thanks.
Thank
Thanks
you.
a lot, sir. Thanks a lot.
Thank you very much.
Nidhu,
So
Dr. Nidhu, continue with the session. Sir, if
may I leave? May I
Sir,
leave?
you are
I have to go. I have to go somewhere. I have already told you that I have an urgent meeting there.
Okay, sir.
Thank you very much.
Thank you very much, and all the best for you.
Thank you, sir.
Now, moving forward with having another plenary lecture, we are pleased to welcome our distinguished invited speaker, Dr. Hemant Nuthlapati, whose work on application of AI-assisted Raman microscopy from single-cell biology to diagnostic will represent a significant advancement at the intersection of the technology, biology, and sustainability. Dr. Nuthlapati serves as an associate professor now at the Raman Center of Medical and Biological Applications, Shimano University, Mastu, Japan. Originally from southern India, he earned his master's degree in chemistry from Banaras Hindu University, Varanasi, and he went on the pursue his PhD at National Chiao Tung University, Taiwan, as a recipient of prestigious Taiwan scholarship. His doctoral work explored the application of Raman microscopy in single cell analysis, an area has since expanded significantly. Now, a postdoctoral fellowship at Ultimate Spectroscopy and Imaging Laboratory in Taiwan, Dr. Nuthlapati joined Shimano University in 2014. His current research leverage AI-powered Raman spectroscopy for the early diagnostic of complex diseases such as cancer, revolutionizing how we approach medical diagnostic. Additionally, he has played a vital role in promoting Indo-Japanese collaboration in education and research, serving as a bridge between leading institutions of both the nations. So we are truly honored to have him with us today. Dr. Hemant Nuthlapati, now you can start your presentation, sir. Thank you very much, sir.
Thank you very much. I just first want to make sure that you can hear me and also you can see my screen.
Yes,
Is it
yes,
fine?
you are clearly audible, sir, and your presentation is also visible right now.
Wonderful. Yeah. First, let me thank Professor Dwivedi and Professor Shukla for giving me this wonderful platform to discuss some of our recent results. So I'm Hemant. I'm an associate professor and also a deputy director for international affairs in Shimano University. And today I'll be talking about what we do in our laboratory. So primarily on applications of AI-assisted Raman spectroscopy. We all have heard, CV Raman, so it's the same name, right? Raman spectroscopy. What in my laboratory we are particularly focused on is studying biological systems. As you see, I am in department or faculty of life and environmental sciences, and especially in the department of life sciences. So we are definitely trying to, understand life, in any way possible using the tools that we have. So as the conference is rightly set up during this very vital time where India is rapidly growing, so it's very it gives me pleasure to be a part of this initiative.
And the pleasure is ours that you are here. We welcome you, and I definitely hope that the participants are going to be immensely benefited by your presentation. And I always look forward for your presentation because last time also when you gave, it was very informative. And I hope the participants will really enjoy as well as learn many things from your presentation. Welcome you, sir. Welcome you.
Thank you. Thank you so much for your kind words. So as you see, for achieving VIKSIT BHARAT 2047, what we are trying to do is focus on number 3, SDGs number 3, right? So that is good health and well-being for all. Because I usually run out of time, I keep talking too much, and then I forget to acknowledge people who have been very kind to me over years or decades, for which I'm very grateful. So Professor Yamamoto, Professor Hamaguchi, and so many collaborators and students, of course, funding bodies. Otherwise I would not be here to show any results at all. Thanks to all of them. So how do I, contribute to the VIKSIT Bharat, in our own way, right? That is the outline I made here, where we are looking at, I said, focusing on SDG 3, which is health and well-being for all. But given my background and specialty, so my idea is to, keep spectral and imaging plus big data as the central core of my own, laboratory, and also try to, contribute in various ways, right? And out of that, how I envisage my career is in these 4 main pillars, right? So we have very much we have a lot of expertise in developing new Raman systems and so on, optics and so on. So we are trying to use it for various applications, not only for disease diagnostics, pathology and pharmacology, food and agriculture, and environmental applications. So I'll give you a very brief look into some of the results that we got in the laboratory in each of these fields. But also, this is actually possible only with the advent of AI and ML. So we have a very nice team, 2, 3 PhD students who are dedicated completely for data science. So where we try to extract meaningful information from the large data sets that we obtain from our laboratory. But that is one half of the idea. But it's also very important because Raman spectroscopy is not a universal solution. It cannot solve all the problems. So obviously, we try to also borrow ideas and also try to integrate other modalities. For example, looking at endoscopy, we would like to put Raman endoscopy, or put Raman plus digital holography, or Raman plus electronic nose, so that we can get complementary information and make much better decisions in terms of or solve the problems. And eventually, in order to really truly realize the potential, we have to obtain a lot of data, which is actually very limiting when it is not automated. So this is one area we have not started yet. To be honest, this is my future for myself. But all the 3 areas we have been already very successful, and hopefully, if we can try to automate it, we would like to really move to the next phase of spectroscopy. So what is Raman spectroscopy? For those of you who have attended my talk last time, I'm going to very briefly also introduce what is Raman spectroscopy. Raman is Sir C.V. Raman, who got Nobel Prize in 1930. He discovered it in 1928. And this is a very high, landmark. Of course, we all should be proud of, just being an Indian. And one very important thing I'd also like to point out is that he was the first Asian Nobel laureate in science, right? So that is not at all, a small feat, especially given that we were under British rule at the time. So anyways, what does it do? What did he detect? What did he discover? He discovered that when light is, falling onto, especially very specific wavelength of light falls onto any material, which is, let's say, all these materials are made up of molecules. So then there is some sort of interaction that is called as inelastic scattering of light happens. And what technically, we are trying to look at is the molecular vibrations. We know that molecules are vibrating all the time. And if we can, somehow, the vibration depends on the kind of atoms that are involved in the chemical bond. So if you are able to measure the vibrational energy, because everything needs energy, the atoms are moving, there is some sort of energy. So if we can measure the vibrational energy, we can understand what are the atoms involved in the molecule, and then eventually we are able to identify the molecule, right? That's a very basic thing. I'll not go into detail because, for the lack of time. How do we actually do, for example, the experiment? Any sort of sample is fine, liquid, solid, anything is fine. Just take a sample, put a laser, and then what we see is if I put a green laser, then light is scattered on all directions. And then even though I give only green light, you can see also different colors of light coming out of that point, right? So different colors, these are Raman light, these are Raman photons, simply as simple as that. What we try to do is to actually collect only those photons that have changed color from, let's say, the incident light that is green here. Then we cut all the green light, collect only the other colors. And of course, it's very, very, very weak process. It's 1 in a million photons change color. So it's not easy. We have to have a very high performance spectrometer and CCD to actually catch it. And then finally, we can make some sort of a spectrum like that. And this spectrum is like a fingerprint molecular. So like our fingerprints, which is very unique for each individual, this Raman spectrum is also unique to each molecule. So that's why we can, totally identify it, with very high specificity. And how we can, also measure very small samples. All you need to do is to actually just put this light or laser into a microscope, right? So you can put the laser into you can use some optics and so on. That's fine. Nothing so difficult. We just try to introduce the laser light into the objective lens or into the microscope, and then you can actually try to observe very small samples like cells and tissues and so on. So similarly, this is a machine that we developed ourselves. The main advantages of these kind of technologies is that we can get sub-micrometer spatial resolution, and we don't have to actually use or destroy the sample, or you just leave it as it is. So right, you don't do anything else. You just put some light onto any sample. That's all, right? And we also don't use any labels or dyes and so on. If you are looking at single cells analysis, usually people have to identify the molecule and put some tags, chemical dyes and so on, which will actually alter the whole system, but we don't have to do that here at all. So label-free technology, which is sub-micrometer spatial resolution and also in vivo, right? So that kind of advantage is huge, especially for biological studies. And what we can also do is, we can do imaging. Right now, it is chemical imaging because we are looking at molecules. There will be molecular imaging. Let's assume that there is a sample like that, let's say a cell or something. And if you divide it into, let's say, 16 points, 4x4 matrix. So what technically we do is it is under the microscope, you see like this, which is about, let's say, 10 microns in diameter. And simply, if you just, shine your laser in a very tiny point, let's say it's 1 micron size here, then you get Raman spectrum, very specific from that particular point. So now what you can do, you can move the laser at different points of the sample and get very, specific molecular information obtained from each point. So that way, it will reflect different molecular compositions from the, different geographies. So now, in reality, we collect thousands of data. And what I show here is actually real data. And this data contains about 6,500 spectra, and each spectrum is about 1,300 points. So technically, we have millions of data points, about 8 million data points here. And that's why we have to use lots of multivariate statistics and data science, right? That's what comes in. And this is a very large part of the research. Of course, experiment is 50%, let's say, and then analysis is about half of it. And we can do all sorts of things. And then eventually what we can do is we can obtain images like that. These are chemical images. Now we don't have to put any labels, as I said. You just put some light, collect the light that comes out of it, and then you do the analysis. Finally, you are ending up with beautiful chemical images. Given that background, let's say these are the facilities that we have built over the years. We have purchased some store-bought machines as well, and so on. So we have different lasers, different spectrometers, and all sorts of things that are required to study various samples. So I'll give you very, very quick, examples, let's say, right? How do we study single cells? I'm going to give you an example of an yeast cell, which we have been very, obtained a lot of, information so far over about a decade. One thing very simple that has not been actually understood very well, because yeast is a very popular model, single cell model for various, biological studies. However, the chemical makeup of the cell walls are not very easy to study. So now what we tried with Raman spectroscopy, because it is composed of various kinds of polysaccharides, it could be glucans, it could be mannans, and these are all not just simple glucans. Glucans may be alpha glucan, maybe beta glucan. There are different kinds of, linkages, 1,4, 1,6, and so on. And also these are all structurally very similar. Only the bond angles, etc., are different. And these are isomers, right? So obviously, other techniques cannot identify easily, right? But with Raman, yes. I said this is going to be very reflective of the structure, molecular structure. So what we did, we tried both, mitosis and meiosis, both sexual asexual reproduction, to make spores. And while making spores, what happens is it makes 4 spores, and then there are new membranes or walls that actually form. These are called as spore walls, and these are the mother cell walls. So now there are a lot of dynamic, cell wall. Reorganization is happening. So it's very hard to understand what kind of chemicals go in there, right? So if you just measure different points of the Raman of the yeast cell, you can get very different Raman spectrum, which is indicative of, let's say, lipids or proteins or DNA or polysaccharides and so on. And then what happens, we can actually run through the library, or we make our own libraries, and then we try to identify what are the molecular markers or spectral markers rather. And then we say, okay, these are the bands that are very, sensitive to alpha glucans or beta glucans and so on. So now we can identify, such spectral markers, and then you can use those markers to actually now generate Raman images or chemical images. And very interesting thing is that when we make images of, let's say, alpha glucans and beta glucans, especially in the case of spores, you can see that even though there are 4 spores inside, the spore walls are only showing alpha glucans are the only ones that are inside. There is no beta glucan observed inside these cell walls. But on the contrary, biologically speaking, people have always been under the impression that beta glucans are the major glucans. So this is very surprising because usually the total content is, probably beta is more, but nobody knows where is beta and where is alpha. And that is why this kind of beautiful, molecular images would be extremely helpful. And, you could also do some sort of correlation imaging, and then you can identify that both alpha and beta glucans are only present in the cell wall, but not inside. So that kind of very detailed information is obtained. But anyways, very important thing is that when we identify these marker bands like this and say this is alpha and beta glucan, that is absolutely fine. But many molecules are very similar in structure and very slight difference, and that may actually lead to overlapping bands. So that means we cannot identify one band for one molecule all the time. So that means what we can try also is to now apply some sort of advanced multivariate statistical models and try to extract. It's like, data mining or instant pattern recognition kind of a thing, where we are now trying to say, okay, I know there is something more in there, but I cannot see it very easily. Now let's try to model various kind of spectra and then see whether we can get some meaningful information, which is not otherwise obtained. That way, what we also obtained is now something very surprising, because there are 2 separate polysaccharide compounds that we could separate as a whole, and these are mannans, right? So one is glucan and the other is mannan, because previously we could not find mannan. Now, after the analysis, we are able to actually find mannan as well. And when we look at the images of mannan, then it's much more surprising, because it is usually, it is primarily observed inside these spores. So the makeup of, the outside is beta glucan and the inside is alpha glucan, and within that we also see, a mannan is very, in fact, for the first time, we could see that, experimentally. So this is, wonderful. So that kind of a very detailed analysis we can do, right? So that gives you an idea of how we can study single cells simply using Raman spectroscopy. And how and where we can apply these things, we can use it for various use cases. Let's say to study in pharmacokinetics or, technically pharmacological applications, or we can also use it for clinical applications in terms of diagnostics. So even given this, let's say the fungal infections are very common. In fact, I'll give you some very recent example. So terbinafine is a antifungal drug, right? This is an antifungal drug. What it does is that it actually, just like we have cholesterol in our bodies, yeast cells have ergosterols. And now this is very important part of the cell walls as well, or the membrane rather. And so this drug, what happens? This drug stops the synthesis of ergosterol, so that making the cell not viable, and that's how it has a drug effect, right? So now, if I just simply, let's say I'll directly go to this one. So just before adding the drug, when I measure Raman spectrum of yeast lipid droplets, it looks like that in blue, right? But I just add the drug, and then the drug actually has the drug has a very nice band around 2200, which is coming from CC triple bond. So CC triple bonds are not usually observed in living systems. So obviously we can use this as a very specific marker for the drug, right? So now, if you do that, what happens? Just as soon as you add, probably about, because the experiment itself, it takes about 15 minutes. So just roughly after 20 minutes of addition of the drug, you are able to actually see the presence of the drug inside. So you are able to do drug imaging, directly drug imaging. That is a huge advantage. All pharmaceutical companies want that, right? They want to see where it goes, what it does, and so on. So we are able to actually see in different cells. You can see that just after addition, you can observe or image drug. And we can also do very detailed analysis in order to see how long the drug is inside the cells. For example, with just 0 hour, there's no drug, and then we add the drug within 30 minutes or so, we have very strong presence of the drug. Even after 2 hours, 3 hours, the drug is there, and then it's gone, right? And then we can also re-add it after 30 hours just to see, and then we can observe the re-uptake of drug into the cells. So the very interesting thing is, as the drug is, becoming effective, you can see this particular band, which is a marker for ergosterol, disappeared completely here, right? There's no band here. You see this band. So that means the ergosterol is blocked. Ergosterol is not formed, and we can see that. So it's not only the drug itself, we can see the drug itself, but also the drug-induced effects can also be observed like that using Raman spectroscopy. And these are very detailed analysis as to what time has and so on. And similarly, we can also visually observe the effect. So the terbinafine is the drug. We can see the drug is like that, and then it's gone. However, squalene is the effect of the drug, right? Because the ergosterol is gone, that means squalene is going to accumulate. And now you see, very interestingly, even though the drug is not there about 8 hours or 12 hours and so on, however, usually squalene is not present in the cells because it just moves to become ergosterol. But because the drug is effective, it cannot make ergosterol. It has to accumulate squalene. And very interestingly, we see the presence of squalene here. So even though the drug itself is not seen, the drug is still very active until about 20 hours or so, right? So that kind of detailed information we can obtain. So another very, so okay, how can we actually apply for diagnostics? Now, this is CML, this is chronic myeloid leukemia, it's blood cancer, and this is a very kind of a genetic, origin where there is an exchange of, BCR and ABL between chromosome 9 and 22, leading to a, cancerous product, gene, right? And this BCR-ABL gene product, the protein, that is what is, the culprit. So anyways, normally we don't see that because they are different. We don't have this active tyrosine kinase product or enzyme. However, what happens is, anyways, once you have this disease, right? Now what we do, we go to the doctor. Doctor will give you a medicine, and the medicine is there, very effective. That is called as imatinib, right? So, so we don't have to look at all the details. Okay. Technically, the drug is imatinib, and what happens is imatinib will go and bind competitive binding, and it will stop the BCR-ABL gene product from signaling, right? Technically, this can stop or not cure totally, but technically the point is it can, work on the cancerous cells like that. However, what happens is if you keep continuously taking this medicine again and again, there is a very good chance, about 10% of the people actually gain resistance. So now we are talking about, let's say, antimicrobial resistance or, so many kind of drug resistance, right? So this is, resistance to cancer drug imatinib. And this is a very serious problem where if you don't identify it very quickly early on, it can lead to a very dangerous phase called as blast phase, where it's impossible to treat at all, right? So now, how do we do that? Usually it's very difficult because normally you have to, go through so much of, you have to take a lot of, experiments and do different kinds of analysis and put together and try to understand what is the situation. However, it also takes a lot of time. So it's very expensive. It requires a lot of, facility. It requires a lot of trained personnel and so on. So it is not very, it's not, available to everybody. However, so what we tried was, okay, that is very standard one. That's fine. What we did was we tried to use Raman spectroscopy and see whether it is possible to differentiate cancer cells. Both are cancer cells, but cancer cell that is sensitive to drug and resistant to drug, right? So, so first, of course, it's a collaborative project with, in fact, Tata Memorial Hospital in Mumbai. So they already did a very nice array CGH analysis, all the genetic analysis they have done, very detailed, and then they found that there is some sort of a chromosomal gains, right? So they found chromosomal gains. That's fine. So that is an indicative of drug resistance, development of drug resistance. So now what we did Raman spectroscopy for both sensitive and resistant cell types. It looks absolutely similar. I mean, but not identical. There are some differences, very small differences that we cannot see with the naked eye. So what we try to do is, of course, we do multivariate statistics and machine learning, right? So we can see that there is some sort of difference between the two groups of data, even though it is not very straightforward to the naked eye. Well, that already gives a very good confidence that we can further go and try to do something with that data. Well, we could also do supervised learning, where we could do linear discriminant analysis, where more than 90% accuracy, we are able to differentiate between these two cell types. But we still don't know why we can actually separate. We can separate, but doctors, we also, as doctors, are more interested in molecular information. So what we did was, now we tried to do this pattern using multivariate curve resolution analysis, where we are trying to extract meaningful, physically meaningful, biological components from these data sets, right? By doing that, what we also were successful is to extract the DNA information as such. And then when you look at the DNA, ratios between the two groups, especially with different molecules, we are able to identify that indeed the resistant types have gained more DNA, right? So now the array CGH is going to give you very detailed, genetic analysis. That's fine, which is very expensive, time taking, and so on, not available everywhere. But even though it is not exactly the same data, but a similar information can be obtained using Raman spectroscopy, which is much more easy to do. And, we can kind of, democratize it, when necessary, right? So that kind of thing, we hope we can translate, use this as a translative technique. Well, there is another, I said I was talking about endoscopy, where we would like to do Raman endoscopy. Why are we interested in it? Because, for example, there is a disease called eosinophilic esophagitis, and that is especially the, infiltration of eosinophils. These are WBCs, right? One type of WBCs into the epithelial layer of the esophagus. Esophagus is the food pipe that when we eat and drink, we feel it, right? That is the place. So normally we don't have any blood cells on the epithelial layer, but with age and I mean, especially in Japan, it is primarily observed in aged population, which is why Japan being a great society, so we see a lot of cases. So, well, we already know Raman spectroscopy is very specific to different molecules. So that means these blood cells, white blood cells, may have different kinds of molecular makeup, and we can identify with Raman spectroscopy. That we already have established previously. So we are now trying to develop that as an application, where we, of course, we cannot directly go and do it with the patients. So we did with mouse models, where we induce this disease intentionally, and then we cut, we sacrifice it, and then we take the tissue of the esophagus and then measure whether it is possible to observe the presence of these eosinophils or not. And in fact, we did observe very clearly using a standard microscopic setup that is very, very good. And of course, we can also, kind of cross-check, of course, with H&E staining. The doctors confirmed that this is also present with the same tissue. Wonderful. But now, what is the catch? The catch is, under this setup, you can, do that. But it is very important. What we would like to achieve is that we want to combine this into an endoscope, where the doctor can put, directly a small fiber and then get molecular information directly from the esophagus, so that we can immediately diagnose whether there is, eosinophils or not, without cutting it. The biggest advantage is that we don't have to do any, we don't have to remove any tissue, right? Well, finally, one small example, this is breast cancers, and how is it possible to separate breast cancers from normal breast cells? So in this case, it's still cellular system, not the cancer system directly at tissue level. So we, of course, again measured with both normal and cancer cells, breast cells. Then it's a very similar story as we saw in case of blood cancer. However, the very interesting point is that we actually got about more than 90%, 98% accuracy for separation of normal breast cells and breast cancer cells. And what is the reason? Especially the reason we found was that there is a very specific lipid type, especially that is a diunsaturated lipid that is very different between these two cell groups. And that is very, very useful, because we know that these are the precursors for prostaglandins, for example. And those prostaglandins are kind of, inflammatory molecules that actually lead to, cancer. So what we are trying to look at is this very nice molecular, markers that may be indicative of many things. But of course, in this case, because we have a very controlled system of cancer and non-cancer cells, so we are able to very clearly identify this difference, difference in the molecular makeup. So, but we also have to actually automate it. And how do we automate it? It only because all the models that I've been showing so far, we have to manually do a lot of things. We cannot, make an automated pipeline. So for that, we are applying, AI, especially using neural networks. What we try to do was, develop a model that can immediately differentiate between cancerous cell and a normal cell. And in fact, it also achieved 98.3%, or at least 98%, without any human interference, right? So now if you give the data to the model, now it will say whether it is cancerous or not cancerous. So we went one step ahead. I'm not showing the details because it's still under review now. These are explainable AI, where we would like to also look into the brain of this neural network to see what are the reasons as to why this neural network is performing so well. So this kind of explainability is what we are primarily interested in. And last maybe 15 seconds, I'll give you some examples. We are looking at, the application of Raman spectroscopy for, making novel materials in collaboration with professors in IIT Jammu. We have come up with some sort of a specific, morphs where we can deliver with high precision drugs to very specific organelles such as, mitochondria or lipid droplets and so on. And we can see that very beautifully. Similarly, this is a very different thing. We are also very much interested in food and, food analysis. As I said, we are interested in integrating different techniques. This is something called as eNose, where we are trying to obtain information about volatile organic compounds, VOCs, and we are able to, differentiate between different kinds of oils. And immediately, that means we can use that for adulteration, detection of adulteration in food, especially in case of oils. Similarly, we are also studying a lot on, microplastics, because it's a huge problem, especially in the case of environment, monitoring. So we can see how the interaction of nanoplastics and microplastics with cells, what happens and how it affects. So that kind of a detailed information also we are working on. And again, there's another example of how we can study starch or, different kinds of, we studied many polysaccharides in yeast, but why not, in rice, let's say, right? These are different kinds of polysaccharides, of course, based on amylose and amylopectin. And we've been very successful in estimating the ratios of amylose and amylopectin in various starch varieties. These are, of course, samples from India. And, this is the way we are working on. And I hope in the future we have some time to visit Japan. Please do visit Shimane. It's a very beautiful place on the west part of the main island, where the sunset is, it's a beautiful sunset place. So it's very popular in Japan, in fact, for sunset. So I welcome you all. Thank you very much for this opportunity.
Thank you, sir, for your nice lecture. And it was very informative, and it has so many things. I think participants would be definitely benefited with this. I request the participants, if any queries there, you can post your queries in the chat box. It's a very informative lecture. Participants, I think that if any query would be there, then we'll definitely post you and
write to me, or you can ask later. Everything is fine. I'll put my email address in chat box, so it's fine. I think I've already taken too much time than allotted, so I am very sorry for that.
No, sir. It's for see, if something is we are getting, the participants are getting, so there is no limit for the time.
Yeah, I'm very glad.
Thank you so much for your lecture. Dr. Nidhi, over to you.
Yes, sir. Sir, thank you for your informative lecture, and thank you for sharing your valuable knowledge and research with us today. Your insights into the intersection of life sciences and sustainable practice are truly enlightening, and I think there are few participants who will ask questions. Kindly raise your hand. There is one participant who wants to ask. Just a second.
Nidhi, I think sir has left.
No, I'm here. I'm here.
Okay.
Yeah. If there are any
Okay.
questions, very glad to answer.
Question chat box right now.
One
Oh.
participant is saying that it's a too good lecture. I just recalled NMR after a long time.
Yeah. Nice.
Okay. Anybody else who wants to question? Kindly raise your hand, or you
Sir,
can
in
write
the
in
meantime,
chat
if the
box as
questions
well.
come, I am really very glad and impressed to see Professor Radhakrishnan's photo in the background of your office.
So, I mean, that is my bread and butter because, I mean, I've been talking about Raman all the time. So, I mean, I also want everybody who visits my office to also see that he's the person. Right.
Now, it's very heartening to see that people, you're working in Japan, but you carry the legacy of Professor C.V. Raman with you.
We all do.
This makes us all proud.
Absolutely.
Right?
Absolutely.
So all the background work related to the Raman.
Yeah.
So,
Yes.
sir, can I ask a single question?
Yes,
So,
please.
as you using some drug like Algerol or some other drug, but can we know what could be the sensitivity of that drug on the detection point of view over the cell? So,
Right.
is it some picogram or nanogram?
Very good question. In fact, each of the drugs that I discussed today is one lecture independently, right? So the point is, while I said there is very good advantage of looking at the drug, the technique that we are using now is spontaneous Raman spectroscopy, where it is not very highly sensitive, right? I mean to say, especially for that particular application where we are giving it, picomolars and nanomolars, we cannot detect it unless there is some sort of accumulation, right? Even if there is a low, see, you administer picomolars or micromolars, whatever, doesn't matter. But as soon as it enters and it accumulates in certain areas of, either tissue or cell or somewhere, if there is a strong accumulation and a local concentration is, relatively high, then at least micromolars and up, not sub-micromolar, it's not possible. And if you want to detect such, then we have to move for different kind of techniques, or we have to, come up with different strategies.
Okay, it means single molecules cannot be detected
We
over the
cannot
surface.
detect single molecule with this technique. With Raman, of course, there is something called as tip-enhanced Raman spectroscopy, and where you can actually see even few atoms, right? Not only the whole molecule. Sometimes that become a very thing. If it's it depends on, but it does not. It is not possible to study living systems as it is, right? So there is both, positives and negatives. We have to, just choose. In this case, particularly if you're interested in drugs to study in living cells, what we can also do is something called a SERS, surface enhanced Raman spectroscopy, where we can try to look at even nanomolars and picomolars. Sometimes it's possible.
So, can you suggest any of the technology which able to detect the single molecule drug over the cell wall?
It's really hard.
Oh, no issue,
So,
sir. But this is a good
I mean, of
technique.
course, if
Actually.
there is some way, for example, if the molecule has, of course, single molecule fluorescence spectroscopy, we can try. That is not a thing. But first, in the living systems, it's it's going to be extremely hard. Maybe we can try with, model systems. We can make artificial layers. I mean, artificial, membranes, and then we can try to, see presence of, some interaction with the drug and, lipid bilayers. I think some people are doing that. But I cannot do that. I have no expertise in that. Yeah.
Thank you, sir. We have learned so much from your lecture. Actually, Raman is really a legacy, as sir said. Thank you, sir.
Thank you very
Thank
much, sir. Thank you for this wonderful interaction.
Thank you.
Now we will now move on to the next segment of our session, which involves contributory papers and presentations. That let me take a moment to introduce the research papers and the authors as well. Firstly, we are having CP55, which is titled as a comparative study of issues and challenges of population growth, changes in land use planning and environmental degradation in agroclimatic zones of Uttar Pradesh, India. Hariom Tyagi, are you here to present it? Now moving to the next one, CP53, which represents the paper title Evaluation of Physico-Chemical Parameter of River Water in the Lower Gangetic Delta region, West Bengal. Arindam Ghosh, by Arindam Ghosh. Arindam? Okay. Now following to the next one, CT54. The paper is having unraveling the influence of social media marketing on expansion of net zero initiative of healthcare, authored by Gaurav Bharadwaj. Anybody who wants to present it? Mini Agrawal, Shweta Saxena, ma'am.
Next.
Okay, we have CP55, which will focus on the study, the effect of seedling of chickpea and lead and cobalt, by Manish K. Yadav. Manish or Arvind, are you going to present it? Anybody? Okay, now moving to the another one, CP56, Eco-Sustainability Study of Non-Combustible Vehicles, Vikrant Singh Chaudhary. CP57, Advancing Biohybrid System, Enhancing Photosynthesis for Sustainable Energy and Agriculture, by Shikha. Yes,
I'm
Shikha?
here, ma'am. Yes, ma'am.
Okay, okay. You can start your presentation, Shikha. Shikha
Okay.
Diman.
I'll just share my screen.
Okay, thank you very much.
Is my screen visible, ma'am?
Yes, it is visible.
Okay. So the topic of my presentation here is advancing biohybrid systems.
Excuse me.
Yes, ma'am.
It's your poster. You can
Yes, ma'am.
please put it on slide mode.
Yes, ma'am. I'll do that.
Yes. Okay. Okay.
Yes, ma'am.
You can start.
So the topic is advancing biohybrid systems with enhancing photosynthesis for sustainable energy and agriculture. So one of the major problems with the photosynthesis machinery is that these machinery can only absorb light for in a specific range of light spectrum. So we have designed biohybrid systems wherein we are taking chloroplast as a biological entity, and also we are taking a metal nanoparticle. Combining the two moieties, we have decided that these two can enhance photosynthesis, as metal nanoparticles are known to absorb light and convert it into excitons. Now then, these excitons can be transferred to the photosynthetic machinery, wherein these electrons can go through the entire cycle and produce more number of ATPs as an final step. Also, this can also help with additional yield of crops, which we have seen in our results. So in our lab, I'll move to the methodology part. I'll just zoom it a bit. Give me a second. Yeah. So for the methodology, we first use the nanoparticles with the chloroplast, and then we have studied the photoluminescence property, whether these are enhancing the absorption pattern of chloroplast or not. Then further, these nanoparticles, these are the nanoparticles that are present over here, and this is the entire photosynthetic machinery wherein we had photosystem 1 and photosystem 2. So these nanoparticles transferred electrons, it was observed that they transferred electrons. There was more splitting of water. This entire system we mimicked in our lab as 5 different reactions. The first one was splitting of water, where we have seen most number of water molecules being splitted. Then we have also studied the oxygen evolution, how much oxygen is being evolved in the presence of nanoparticles. Then we have studied the reduction of ferricyanide, and all these reactions we performed in presence of artificial electron donors. Then we have studied the production of NADPs and ATPs in the entire cycle. Then we also started the uptake and translocation study of these nanomaterials within the Arachis hypogea plant system in vivo study. Then we did the toxicity analysis and the plant growth analysis, where we have seen the physical morphology of these plants. The very first step was the isolation of chloroplast. So we isolated the chloroplast and treated it with different concentrations of the metal nanoparticles. Then after that, these are the characterization and synthesis and screening of these nanoparticles. As you can see from the photoluminescence spectroscopy, these nanoparticles, in the presence of nanoparticles, the peak at 680 enhanced, which clearly shows there was more electrons being within the system being produced. Also, one of the advantages of using silver nanoparticles was that they were the one that we synthesized in our lab were autoluminescence, and that luminescence property also contributed to higher number of electrons. Now, the effect on photosynthesis, light-dependent reaction was studied, including the DCP-IP reduction, ferricyanide reduction, oxygen evolution, NADP, and ATP synthase. And it can be clearly seen that with increasing concentration of these nanomaterials, we can see higher amount of DCP-IP being reduced, ferricyanide being reduced, oxygen being evolved, and NADP produced, as well as more number of ATP being produced. Now, from the uptake and translocation study, we can see that the nanoparticles were seen within the root, petiole, and the leaf sections of these plants. Now, the first image, the A part shows the fluorescence microscopy images, whereas the B part shows the confocal microscopy images. And from the confocal study also, it can be seen that there was a presence of these nanoparticles. Now, these nanoparticles were seen in green color because we have tagged these with a fluorescent dye. Now, we further studied the toxicity and plant growth. So we have incubated these plants and grown them with artificial cycle of 10 to 14 day and night cycle, maintaining the humidity in the plant growth chambers. And it was observed that more number of leaves, improved shoot length were observed for nanoparticles, in presence of these nanoparticles. We also did the MTT study where very less toxicity effect was observed, that too for the highest concentrations. Finally, moving on to the conclusion, it was seen that these metal nanoparticles can absorb light for the broader range of UV visible and near IR spectroscopy. We have worked with a few more nanoparticles. We have worked with CQD. We have worked with the core shell nanomaterials, where we have shifted we have redshifted the absorption band of these nanoparticles. Then we have also studied it with the MWCNT, TiO2, and other materials. And we have seen an improvement and augmentation in the photosynthesis machinery and the flow of electrons for the entire thing. Now, since we are harnessing the power of these photosynthesis for environmental solutions, we can apply them to the biofuel production, artificial photosynthesis and energy conversions, and for the storage. Also, later on, these can also help the plants to grow in minimum intensity of lights, where less light is visible and available for the plants to absorb. Also, in stress condition, it can help. Also, these nanoparticles have certain more properties where they help the plant to be resistant to certain disease. They can be resistant to the drought conditions and hold more water capacity. So, all in all, these nanoparticles can also act as nanofertilizers. Simultaneously, they can enhance the light absorption, enhance the photosynthesis production, and ultimately lead to the enhanced yield of plants. Thank you. These are the references and the work that I've published with the ACS Omega and Bionanoscience. And I would like to thank Amity Institute and CIRF specially for giving me all the instrumental facilities to be done with this work. Thank you so much.
Thank you very much, ma'am. Thank you. Now I want to ask you questions.
Ma'am, can I just take 1 second? Yes, ma'am.
Yes. Can biohybrid system be adapted to improve photosynthesis in low light or indoor farming setups?
Yes, ma'am. Certainly.
Okay. And how can we ensure the long-term safety of the metal nanoparticle in large-scale farming?
Ma'am, we are working on that part, but for smaller aspects, I can tell you we have observed peas plant growth, peanut plant growth, and until the fruiting stage. So I have seen there was no detrimental effects that we observed,
Okay.
and there were the entire thing. The plants that we grown were equally healthy, and there were no much issues with these plants, and they can be grown easily. But
Okay.
we haven't seen it in a large scale agricultural land, but we have seen it up to 27 days.
Okay. And what role do metal nanoparticle basically play in boosting the electron transport during photosynthesis?
So basically, if you'll see these nanomaterials, they um, absorb the photons and they convert it into excitons. Now, this is exactly what the chloroplast machinery is doing. They convert the light energy into electrons, and these electrons are being transferred. Now, ultimately, these electrons help in bifurcating this part, like splitting of water, where we produce hydrogen and H atoms, and these are further transported. The electrons are being transported through the ETC, electron transport chain, and these are producing ATPs and NADP, which are required for the light independent reactions and production of glucose at the final stages. So we have also studied the carbohydrate, protein, and lipid presence. And we have also seen that the carbohydrate content has increased
Okay. That's
at the final stages.
okay. Thank you very much. Thank you
Thank
for
you, ma'am.
the poster presentation.
Thank
Now
you, ma'am.
we are moving ahead to the another presentation, CP58, evaluating the role of strategic communication and public relations in Swachh Bharat mission from awareness to action. Padmavati Ashish Sharma. Anybody who wants to present it? CP59, role of media in promoting sustainable technologies in India. Okay, moving to the next one, CP60, sustainable business models, how startups are driving green innovation in India. Ritambra, ma'am. CP61, polymer nanocomposite and their thermally stimulated depolarization current properties, synthesis, characterization and applications. Harish Kumar Sharma. Okay, CP63, sustainable agriculture through plant probiotic bacteria, growth protection and yield enhancement. Sonali Sharma. Next one, CP64, growth dynamics and salinity adaptation of freshwater microalgae, insights from the Gwalior region. Ekansh Agrawal. Okay, moving ahead. CP65, organic farming. Swati Yadav. CP66, collaborative sustainability, partnering with NGOs and universities for VIKSIT Bharat. Raj Amritam. CP67, echoes of the environment and eco-critical analysis of Kiran Desai's inheritance of laws. Manu Chandel. CP68, hunger must be satisfied first, only then can development follow. CP71 ketostan encapsulated anti-larval essential oil nanodispersion for Anopheles mosquito control. Vijay Lakshmi Ghosh. CP72 fabrication and characterization of biocomposite film based on uh, gorgum ketostan polyvinyl alcohol, tannic acid, and silver nanoparticle for food packaging application. Pallavi Mandi. CP73 sustainable architecture material and technologies promoting eco-friendly building practices. Aditya Bhattacharya. Impact of Swachh Bharat Abhiyan on school sanitation, CP74.
Hi, am I audible?
Yes, you are clearly audible.
Yes, ma'am. I'll just share my screen once and please let me know if it's visible.
Okay.
Is it visible to everyone?
Yes, yes, it's visible right now.
All right. Good morning to everyone. My name is Adit. I'm a third-year electronics student from Guru Nanak Dev Engineering College, Ludhiana. The title for my review paper is Impact of Swachh Bharat Abhiyan on School Sanitation.
Okay.
So I'll just start. So for the entire presentation, these are a few keywords that I'll be using. SBA is for Swachh Bharat Abhiyan. SBM is for Swachh Bharat Mission, Hygiene, Sanitation, and ODF. So the abstract. Our paper, basically, it's a review paper, and we are highlighting all the developments in the last decade, that is from 2014 to 2024, in areas corresponding to school sanitation and for the rural population. We are also emphasizing on the implementation and the impact of SBA on Indians as a whole. So first of all, a small introduction. Swachh Bharat Abhiyan was launched on 2nd October 2014. It is a nationwide cleanliness campaign. It was initiated by our Prime Minister Narendra Modi, and we have a few aims of the SBA, which I'll discuss in detail. The mission was divided into two subparts. The first being the Swachh Bharat Abhiyan for Gramin, that is for the rural areas, and the next being for the urban, that is for our urban areas. The inspiration for Swachh Bharat was taken from Mahatma Gandhi's vision of a clean India. The vision which he had stated was to declare India ODF, that is open defecation free. And it has also been recognized as one of the largest sanitation programs by UNESCO. So coming to the objectives of SBA, what Swachh Bharat aims to do is it aims to eradicate open defecation across India through access to toilets for all. And with this point especially, our focus has been on the marginalized communities and the rural areas. Secondly, it has to promote cleanliness, hygiene, and sanitation, ensure scientific, solid, and liquid waste management, bring behavior changes through awareness campaigns. Now, this point is very important because, as I'll be discussing further, in the past decades, there have been way too many sanitation programs. But the reason why Swachh Bharat stands on top is because of the behavior changes that it was able to bring. Psychological points like these are very important in ensuring the success for Swachh Bharat Abhiyan. Focus on school sanitation by ensuring functional toilets in every government school, promote hand washing habits and personal hygiene, empowering local bodies and panchayats to manage and maintain sanitation facilities. Now, for this point, in areas where entertainment sources, news sources, and radio, they're not very popular, the only form of government that can actually make a difference are the panchayat and the local bodies. So empowering them has been a very important point. And creating employment opportunities linked to sanitation and improving the quality of life in general. Now, a small timeline. So, as I previously stated, the campaign was launched on October 2nd as a tribute to Mahatma Gandhi. A budget of 1.96 lakh crores has been given for SBA. Our infrastructure focus on toilets, waste management, and school sanitation. On 2019, the vision for which this campaign was set was actually achieved, and India was declared ODF, that is open defecation free. One of the reasons we were able to promote this thing in such a short time, 10 years in the span of human history is a very short time. So one of the reasons we were able to do this quickly is because of the use of social media, entertainment. We all watch Kapil Sharma, we all watch YouTube. We have been looking at posts where people mention time and time again to keep our country clean, to keep our country, to improve the sanitation levels. UNESCO and World Bank also praised the government for improving SBA. Now, the key implementations, as I was talking previously, we have two different sections. One is for the rural areas, one is for the urban areas. The one for the rural areas is led by the Department of Drinking Water and Sanitation, and the one for urban areas is led by Ministry of Housing and Urban Affairs. The majority focus for the rural areas was given on behavioral changes, and for urban societies, more of door-to-door garbage collection, waste segregation, and public toilet constructions were emphasized on. Now, as stated by UNICEF, in 2024, over 4.2 lakh toilets were built in government schools. WASH programs, which stand for Water, Sanitation, and Hygiene, were encouraged to better educate the students about hygiene. Now, from the papers of the last decade, that is from 2014 to 2024, a lot of the work has been done. What these papers, they basically talk about is different aspects of SBA. For example, for surveys and cross-section studies, public perception has been explored, student attitudes and hygiene awareness, they have been explored. Statistical and regression analysis, the gaps between the rural and the urban sanitations have been studied. Now, one of the reasons why even we have written this paper is to study this gap. A lot of the paper, a lot of the stuff that's been on the media, that's been on the entertainment sites, that is places which are more open to the common man, they focus more on the urban side of things. So our paper, we have kept the study more focused on the school sanitation for rural students. Next, we have, of course, the related work that's been done by the papers. So what we can conclude from the papers is that Swachh Bharat Abhiyan definitely improved the sanitation infrastructure and the awareness across India. By 2nd October, of course, as I've previously stated, India was declared as ODF, that is free from practicing open defecation. What these studies showed was that just by building infrastructure alone, you can't really make a change in our country. What you need to do is you need to work on people's behavioral skills. So that is what SBA did this time. We had sources like entertainment, we had campaigns, we had stuff going on Twitter, we had ads coming up on newspapers. So stuff like this helped all people, that is people from all walks of life. Now, what is the future scope of Swachh Bharat Abhiyan? We haven't really been seeing a lot of it on the news lately because it's been instilled into our minds that yes, this thing does exist. What the government is planning is for a Swachh Bharat Mission 2.0 that focuses the shift towards sustainability and digital innovation with, of course, we have conferences coming up, we have technological innovations coming up. So it's better that we keep changing ourselves with the latest updates in the society. So that's been my paper. If there are any questions, I'll be more than happy to answer.
Okay. Good presentation, Adit. Now, I want to ask one question. What indicators are most effective in assessing the long-term sustainability of sanitation infrastructure introduced through any kind of initiative?
Sure, ma'am. So, ma'am, for long-term initiatives, I'll take the simplest example of Kapil Sharma show. We all watch that show on Sony TV, right? So, I believe in order for us to sustain it long-term, what we have to do is we need to work with people's mind. What the Kapil Sharma show did and versus what newspapers did. They're both basically giving the same information. But the reason why we related more with Swachh Bharat Abhiyan as the thing came up on the show is because we could relate with the person. When the person is making jokes, he's saying something relatable and then he's addressing the issue. So the idea was instilled better into our minds. So I feel like psychological changes are very important. And that is one of the examples that I've just stated. Ma'am, secondly, I believe one thing that does lead to long-term is addressing the youth. And the government did this very well because we had trends on Twitter that were trending for quite a while. We had Instagram posts, we had reels. Well, reels have started nowadays, but we had back then we had posts which were being shared a lot. We had funny ads and campaigns for Swachh Bharat Abhiyan. So I feel like just not for the rural places, even for the urbanized societies, even the youth was taking participation in the activities. I remember in my society, we had cleanliness drives all throughout the weekends. Every Saturday, every Sunday, and I'm pretty sure all throughout the country, we had these practices being followed. So working with the minds of the people, that is instilling into the minds, and working with the youth, I believe in today's time are the two important factors. Yes, ma'am.
So what policy or structural gaps still exist that may hinder the equitable implementation of school sanitation program nationwide?
Ma'am, I believe I might not know the answer to this question. But what I do believe for school sanitations is that even though the data that we have calculated from the last decades, we can't be very sure of the fact that yes, if a local body has stated that our village or our city is completely following the sanitation practices, we can't just believe them, because a lot of the time, and as we've seen in the past, governments tend to sometimes not give the exact information just to get the recognition. So I believe what we have slowed down with right now is we have slowed down with the intent that we were going. We have slowed down with our pace. We have slowed down with the intensity. I believe getting back into the same mode as we were in 2014 is what could probably fill up the gap for school sanitations. Yeah. Yes.
Thank you so much.
Okay, I have one question.
Okay.
Yes, ma'am.
That's one question, please.
Yes, ma'am.
So, for regression analysis, what type of software you have used?
Ma'am, for regression analysis, I didn't use the paper since the paper is a review paper. So it was followed up on the data that was what we can say calculated by the people who wrote the paper. But if I think if I was to make a guess, softwares like MATLAB and other such, I believe, technological softwares were used. I am not sure of the exact one.
Okay, I have seen your presentation. I
Yes,
thought
ma'am.
you have done all these regression analysis. That's why I am asking you a question.
No, ma'am. Actually, I show the PPT again if that's of any use. I had written the reference number as well. So the people who did the actual data and who did the actual, yeah. Yes, ma'am.
Okay. Thank you very much.
Thank you.
Now
Thank you.
moving.
Any more questions, ma'am?
No.
All right.
That's enough. Now moving to the another CP75 advances in photocatalysis using tin sulfide nanoparticle, a sustainable solution to dye removal. I think it was offline.
Yes, ma'am. I have presented yesterday offline. Yeah.
Yes. So moving to the now, okay, there are all these presentations have done. As we near the end of our session, I would like to thank all of our presenters for their valuable contribution. And I want to give the special thanks to our chairperson, Dr. Rakesh Bhutiani sir, for their support and guidance throughout the session. And we have now reached the end of the technical session 4 of day 2. And thank you to all the attendees for joining us online and for your active participation. We hope you found this discussion insightful and inspiring. Now, please stay tuned for the next 5th session. We look forward to seeing you all of you again. Thank you and have a wonderful day ahead.
And just want to say that all the participants mandatorily join the valedictory session.
Yes.
It is mandatory for all.
Yes,
Whoever yesterday attended, today, everyone should attend. After the technical session.
After the 5th technical session, which will be preceded by Dr. Abhishek Bharadwaj, sir.
Okay, thank you, Nidhi madam. So all participants should be stay here. So we are going to start a new session, that is the technical session 5. So I request to the Asutosh sir, may we sir have joined or not? Asutosh Tripathi sir, am I audible to you? Most welcome, ma'am. Santosh Bukal ma'am, most welcome.
Good morning,
So
sir.
we are just waiting for the Professor Asutosh sir. So please wait.
Okay.
Hello, sir.
Most welcome, sir.
Good morning. I hope I'm audible. Na?
Yeah, sir, you are audible.
Audible and visible. Okay. Good morning, Dr. Abhishek.
Good morning, sir. Good afternoon, sir. Sir, so you are the chairperson here. So I'm just going to introduce Professor Dr. Ashutosh Tripathi, sir. So Professor Ashutosh Tripathi, sir, will chair the session. So Professor Ashutosh Tripathi, sir, is presently working in the professor and a head at the Department of Environmental Science, Nagaland University, headquarter Lumini, Juheroto, Nagaland. Professor Tripathi has active teaching experience from since 13 years. He has completed a PhD from Biochemical Engineering, IIT BHU. His research interests revolve around the environmental pollution and abatement. He has more than 50 research papers and review papers in reputed journals, and so many books he is edited, more than 20. So he has completed SCRB sponsored CRG project on a municipal solid waste management, is currently running two externally funded research project of Jal Jeevan Mission and climate change in Nagaland. 5 students have been awarded doctoral degree under his supervision, and his student and 6 students are at present working for the PhD under his supervision. He is an active member of the various scientific societies and organizations. He served as a reviewer of the many prestigious reputed journals. Professor Tripathi has participated in several international and national seminars, symposia, conferences in India and from the international. So he has been recently awarded Best Academician to 2024 by University of the Putra, Malaysia. Ranking global ranking 158 it has. So he's honored as a senior scientist 2024 awarded by the Environmental Science Department Association, ISDA, New Delhi. Professor Tripathi has been granted with a 12 patent till now from the Patent Office, Government of India, UK Design, the Indian Design, Federal Republic of Germany, and South Asia, Africa. So thank you, sir. So I welcome to chair this session. So
welcome, sir.
Thank you, Dr. Abhishek. Thank you, sir. Welcome. Namaskar.
Namaskar, sir. Namaskar. And it's very nice to have you here. And
same here, sir.
I'm
very much thankful.
Yeah, I just wanted to share that I had all my childhood in Nagaland, Gaspani.
Oh my God.
Actually,
okay, great, sir.
Yeah,
great,
my father was a serving scientist in ICR Research Institute, which was located at Gaspani, Mejifema.
Okay, great.
And
that's great.
Yeah, so I studied in my primary education up till 8th. I had from Nagaland.
Okay, great.
So it's a bit nostalgic for me to hear.
So same feelings are on the both the side.
Right.
I have spent my youth with Amity, and now I'm spending my this adulthood, you can say, or post youth age in Nagaland.
Yeah,
so
you
were
in the Noida campus, sir?
Yes, sir. I have been there for more than 11 years.
Oh, yes, that's great, sir.
So
and
that's just like a homecoming for me and for you also.
Yeah.
Since I am sitting in this Juhanbotto district.
Yeah,
it's nearby Mugukchung, sir.
Yeah, we visited
nice to hear this.
We visited Mugukchung, Food Zero, and all these places with my parents. Yeah.
Great. That's a great feeling, sir.
And I have a very regular and interaction with the head department of environmental sciences, Mizoram, Professor BP
Mishra ji, BD Mishra, na?
and OP Tripathi ji.
Yeah.
So they are very regular at our conferences and other.
Yeah. I am also in very touch with them since they belong to my senior. They are from BHU.
Yeah.
I did my PhD from IIT BHU only.
Yeah,
So
yeah.
they are in regular touch and they are very good friend or senior, you can say, just like
they are senior colleagues. We are young in comparison to what they are and what they have achieved. Right.
Definitely, definitely, sir.
Okay.
So I am very much excited and thankful to the whole organizing committee that you have given me this opportunity to interact with the audience of the August gathering that we are having. So nice of you, sir, and thank you very much for giving me this chance or invitation so that I can interact with each other. I know most of you are known to me. Sometimes I might not have met, but I am aware of each and every one.
Right. Please, sir, please continue with your lecture. And
okay. So let me just try to start because it has been long since I worked on this Zoom. Na? I'm just trying to share.
Yes.
Let
Oh,
me
sorry,
check.
sir. Just I'm make you panelist. Just wait.
Okay. Okay. Is my skin visible?
Okay, it is visible, sir.
Okay, great.
Quite visible,
Just
sir.
give me one more minute.
Please do in a full screen, sir.
Yeah. I'm doing that. Is it okay?
Yes, sir. It is visible, but it's still not in a full screen.
Okay, I have done from my side. I think there might be due to internet. Na? We can wait for one minute.
Okay, sir.
Internet is always a challenge in this hilly terrains. Na? That might be due to
sir, the things have improved a lot now. When we were there and when landslides used to take place, that are a common feature in the Northeast. We used to get 15-20 days of newspaper in one consignment.
Oh my God. So it's so much improved.
Yeah, it's much improved. The only airport at that time that we had was Dimapur.
Okay, right now one Dimapur and one in is Jorhat, sir.
Yeah, no.
And one another thing that I would like to share with all of you guys, since we are from the same family, you can see at my picture, still I am having the founder president, sir, with me.
That's great, sir. That's great.
His motivation, the platform that he has provided, it is because of him only that I am sitting over here.
Definitely, sir. MIT provides the best platform for us in terms of pursuing our research and our
Definitely, sir.
collaborations and all that. Right, sir.
Definitely. And he has given a wonderful platform and we have utilized that. And with his motivation, right now I'm here. So is it visible now in full screen, Dr. Abhishek? Not yet.
Sir, it is not a full screen actually.
I have already done from my side.
Sir, please try once again.
No, it has been done. I have already done it. From my side, it is full screen. I don't know why it is. The slides are moving.
Sir, click F5.
F5. Let me minimize them. Yeah, now it is minimized. F5, yeah. F5 is also done. Let me just try another network once. No? Might be due to the internet. Just a minute.
Sir, kindly rejoin, sir. Possible. Or you can start. No issue. Okay, sir, are we audible? Okay, sorry, sir, we hadn't told it. Okay, hope you can share, sir.
Yeah, let me try again. Is slides are visible. Now I have to make it full screen.
Yeah, sir, it is visible, sir. It is visible. Just you can start.
It's okay.
Yes, sir, it's okay.
Shall I start?
Yeah.
Okay. A very good afternoon to each and everyone present over here. First of all, I would like to thank the organizing committee to give me this opportunity so that I can interact with the August gathering and be a part of this.
Excuse me, sir. Your screen is looking, but nothing is there. It is blank on my screen.
Yeah. Now?
Yeah, it is visible, sir. You can start.
Can I go in this format only?
Yes, sir.
I think by so I'm very much thankful that you have given me this opportunity to be a part of this international conference on sustainable development for VIKSIT Bharat. Today, one of the topic that I have chosen to interact or to discuss with the young generation, that is plastic pollution, that all of us are aware that this plastic is basically becoming one of the major menace to the society. And we will be understanding the crisis and we will like to try to explore the solutions that we have got. The first thing that everyone must be aware that since its inception, you can say in around 1870, for the first time a polymer was being synthesized artificially. And this artificially synthesized organic polymer, which is made up of the smaller monomers, it has got tremendous applications. There were several advantages which made us to use this plastic owing to its various properties like lightweightedness, then you can say adaptability, it can be molded in any shape, various colors could be added, long lastedness. These were some of the properties which has made this plastic to be one of the most useful useful device for various applications. But eventually what happened with excessive use of this plastic, its production has enhanced, and due to its long nature of non-biodegradability and long-lasting nature, it started becoming as a pollutant. It has been observed that according to 2020 Plastic Europe, it has been observed in 2020 only more than 367 million tons of plastics were produced. And there were different forms of plastics in bag form, you can say in bottles, in wrapping material, in several forms these have been used, and they are found in various water bodies, including river, lakes, wetland, ultimately reaching to the oceans. And they are not only found in the water body, they are present in the atmosphere, the terrestrial environment, even in the groundwater. And this is the marine litter, which is causing a lot of problem, which is being produced either intentionally or sometimes accidentally also, and that is found floating in the bodies of water as well as the ocean. Now, owing to the seriousness of this problem, research related to plastic has nowadays become an epicenter during this particular period, the era that we are in. Some of the basic properties that I have already mentioned, that there are several important properties like non-biodegradability. It remains, due to its lightness, it remains floating in the water bodies. It has got high tensile strength, and it is being ingested and entangled by the aquatic organism. And these are the reasons why it is causing a lot of problem. And what it has been observed that more than 80%, even sometimes 90% of the plastic pollution, especially in marine water bodies, is land-based. And there are various anthropogenic artifacts that I have already discussed that can become marine debris like plastic bags, balloons, bottles, etc. Now, talking about plastics and its relevance with the SDGs, that we are having 17 sustainable development goals are there. So there is relevance with most of the SDG that plastic is being involved. I would like to just give some examples of this SDGs, which are very much important for plastic and its sustainable management. For example, if you talk about this plastic SDG 14, that is conserve and sustainable uses of oceans, seas, and marine resources. Definitely, this proper plastic management, it can reduce the amount of plastic in the ocean and mitigates its effects. Then SDG 1, that is in poverty in all its form. So definitely natural ecosystems, they are vulnerable to plastic waste, and they also have negatively effect on ecosystem services and livelihoods, especially in coastal communities. So there also this for SDG 1, plastic also plays a very important role. Similarly for SDG 2 also, plastic in the soil, they are not only a threat to soil health, soil fertility and crop production, but also to the food security and human health. Then the other SDG we have got, SDG 8, which require decent work and economic growth. Definitely proper plastic management can ensure a decent work and economic growth. SDG 11, sustainable cities and communities. By managing plastic waste in urban areas, we can achieve this SDG 11 as well. SDG 12 is responsible consumption and production. So definitely by reducing the plastic waste and its proper management, we can lower the consumption of waste and we can ensure resource efficiency and circular economy can be also achieved. SDG 13, climate action, all of us are aware that these plastic releases harmful greenhouse gases whenever it is being decomposed in landfills, or you can say it is being incinerated. It releases a lot of gases. As per the global data that we are having, it has been found that at least 79% of this plastic is being simply left in this, you can say, landfills, and only 12% uh, is being recirculated, and rest of them is incinerated. Then it is also found to be responsible for Sustainable Development Goal 15, that is life on land. So definitely we are aware that this plastic pollution not only create problems in the aquatic ecosystem, it also creates problem in the terrestrial ecosystem, and thus this is also integral part of our SDG 15. Therefore, by achieving this sustainable development goal, we require effective plastic waste management policies, practices, and initiatives that can tackle the challenges associated with this plastic pollution. And we have already observed the extent of problem that this plastic pollution is having on the society. Yeah, this is now coming to the theme of this microplastic. This plastic has been divided on the basis of various sizes. Macroplastics are plastic, those which are having size more than 25 millimeter. Then we have mesoplastics having size range from 5 millimeter to 25 millimeter. Here comes the microplastic, which is basically ranging from 1 millimeter to 5 millimeter. We do have mini micro, 1 micron to 1 millimeter, and then nanoplastics, we can say, which is less than 1 micron meter. Now, there are types of microplastic also, and they have got different shapes as well. First type of microplastic is our primary microplastic, which is produced in the same size. That means they are produced in the micro range only, that is from 1 millimeter to 5 millimeter, and they enter the ecosystem in their micro size. And the examples of these microplastics includes fibers, which are generally being released from the clothes that we are wearing. From after washing them, these fibers are being released, which is an example of microplastics. We have got pellets of plastics, which is generally known as noodles as well. That is also an example of microplastic. And these micro beads, which are essential feature, which are an integral feature of these cosmetic products nowadays, that is also an example of microplastics. Then here comes another one, that is secondary microplastic, which is not originally produced in the same size, but they result from the breakdown of larger plastics in the environment, and they, with the action of you can say light, wind, as well as sunlight, and thus they get fragmented, they get converted into shapes like films, fragments, and foam. Then these are some of the sources of microplastics, or the anthropogenic sources that we can say, which are mainly responsible for this microplastic pollution, especially in the aquatic ecosystem. They ranges from packaging materials, fibers containing dress that I already mentioned, that is a microplastic, noodles from the industries, which are pellets or the raw material from which various plastic materials are being synthesized, and then the personal care products that I was discussing about, which releases microbeads, and ultimately they found themselves and found their fate into the aquatic ecosystem. This is a representation that you can see that growth in global plastic production. We have taken a data from 1950 to 223. It has been observed that in 1950, the amount of plastic production was only 1.5 million tons. But in 23, it has been observed to be more than 410 million tons. So we can easily observe the tremendous increase in production of these plastics, and ultimately they are finding their ways into the ecosystem. It may be atmosphere, it can be hydrosphere, it can be the lithosphere as terrestrial ecosystems as well. And these are some of the pathways of these microplastics, especially in river ecosystem or aquatic ecosystem that we can say. And as you can see, there are certain examples like wastewater treatment plants. We have got landfills, industrial effluents, construction works, even from agricultural farming activities, fishing, nets, etc., household activities. So they are ultimately reaching to the water bodies, and within the water bodies, like river, they are also undergoing physical, biological, and chemical changes, and due to which they are further getting fragmented into smaller particles, and ultimately they are affecting the riverine biota, which they are accumulating them, they are eating them as a in you can say in getting mistakenly as food material. Not only that, it has been observed that these microplastics, they are also leaching down or reaching down to the into the aquifers, that is groundwater, and they are also affecting the sediments as well as the forest ecosystem. And ultimately, these microplastics, they are reaching to the oceans. And sometimes even the water that we are drinking, the tap water or the drinking water, it might contain these microplastics. So you can have an overview that from how many ways these microplastics are entering into our ecosystems. And when we talk about the impacts of this microplastic in ecosystem, so definitely it has got several impacts, like it changes the properties of soil. It has been found that whenever this microplastic is being absorbed on the sand, it can change the permeability as well as the temperature of the sand, and which could have impact on sea turtles, which have temperature dependent sex determination. That is one problem. This microplastic can act as a vector for harmful pathogens, because smaller the size, the larger the surface area to volume ratio will be there, and that allows more microorganisms to attach to one piece of plastic, and thus it acts as a vector for large number of harmful pathogens. Then it acts as a attracting center, you can say, or nuclei, or a magnet for toxic materials, especially if we talk about this persistent organic pollutants or POPs, which are basically produced in the agricultural activity, and they can get adsorbed over this particular microplastic, and the concentration tends to get enhanced over them. And ultimately, these microplastics could be ingested or inhaled by the living organism, the aquatic flora and fauna, as well as the terrestrial animals. They can inhale that microplastic, and along with these microplastics, large amount of toxins are also being carried into their bodies. This is the problem related to them. There is one research which denotes that in 93% of the bottled water of 11 brands across 9 countries, they were having large amount of microplastics. Some of the famous brands are like Nestle, Bisleri, Aqua, Epura, Aquafina. You can see the concentration is also being mentioned over there. Even these branded water bottles, they are having large number of microplastics present within them. So this is one example that it clearly shows the presence of microplastics everywhere. Now, this is one of the graph which clearly indicates that these microplastics, they are temperature dependent also. Here we have used certain plastic bottles like PET, PP, and polyethylene is there. It has been observed that at minus 18 Celsius degree as well as 45 Celsius degree, especially in case of polyethylene terephthalate, maximum number of microplastics are being released. Okay, so this is one important factor. There are various cancer risks also being found associated with these microplastics from the plastic bottles. And here also we have taken basically 3 plastic materials: polyethylene terephthalate, polypropylene, and polyvinyl chloride. and different temperatures are being mentioned over there, and for different products. We can clearly see that for water in polyvinyl chloride at a minus 18 Celsius degree, maximum cancer risk values are being observed. Even for water at 4 Celsius degree in polyvinyl chloride, we have observed certain cancer risks. And in fizzy drinks also, in polyethylene terephthalate as well as polyvinyl chloride, even for water at 45 Celsius degree, both for PET and PVC, this cancer risks have been observed. So this is one of the problems which is being found associated with this microplastics, and the major sources, some of which we have already discussed, related to plastic in rivers and lakes. They are basically urban runoff, industrial discharge, tourism and recreational activities, and last but not the least, the inefficiency in waste management failures. So this is something that we have to be aware of. What are the major sources which are leading to plastics in rivers as well as lakes? This is just a representation of the way these microplastics are being transported to ocean water bodies. So this hydrodynamics of microplastic, it is basically being governed by some of the characteristics like hydrological characteristics, watershed pattern, some physical properties of microplastic, hydrodynamic behavior of microplastics, and certain phenomena like buoyancy, gravity, and drag forces. You can clearly see that I have tried to explain two conditions during flood condition as well as during dry season. During dry season, these microplastics, they can be transported via rolling, sliding, and then they get deposited. During floods, there is translocation, deposition, buoyancy is there, and with the help of suspension also, they are being transported from river ecosystem to the ocean ecosystem or marine ecosystem. Okay. Then talking about microplastics in groundwater. So definitely it has been observed that even groundwater has been contaminated with this microplastics. But collecting samples from this microplastic is very much challenging. So thus this determination of the occurrence of microplastic in groundwater is still limited. And we do require more research to discover how this soil property, the rainfall pattern, the changes, land use changes in the surface area, they can affect the percolation of microplastics through soil to the groundwater or to the aquifers. So this is one area of research which is very much important. Another area of research which is now being taken into consideration is its microplastic interaction with metals as well as plants. The heavy metal absorbed microplastic, it can increase phytotoxicity in the plants, and thus we require more emphasis on coupled effect of heavy metals and microplastics on the plants interaction. You can see clearly in the picture that it has been described that these microplastics interact with plants, and they can have adverse effect on various activities, the biological activities, physiological activities related with plants like photosynthesis, respiration, the plant growth, presence of chlorophyll biomass as well as oxidative stress. They get accumulated in the soil along with the heavy metals, and they can be translocated, and they can even reach to the fruiting parts or fruiting bodies of these plants. And once they are being inhaled by human body, it can have various problems on human health. It can lead to DNA damage, there can be oxidative stress, and even it can lead to development of cancers in various parts of our body. So this is one of the problems which is being associated with them. These microplastics, they are not only found in aquatic ecosystems, they are also being observed in terrestrial food chains. They contaminate the soil and they get bioaccumulated over there. They can be absorbed by the plants, and since not only they are bioaccumulated, they undergo biomagnification as they are non-degradable in nature, and thus from plants, they can be transferred to human food also, and thus it can affect human health as well. Okay. Some figures related to this presence of plastic in the ocean ecosystem. It has been observed, according to certain survey, that 33 billion pounds of plastics enters the ocean every year, 75 to 199 million tons of plastics are found in our ocean. 70 to 80% or even more than 80%, sometimes even 90% of plastic in the ocean water, they comes from land-based sources. And basically there are 5 garbage patches which is currently floating in our in ocean. And one of the largest garbage patch that we are having is Great Pacific Garbage Patch, which is located between Hawaii and California. All of the environmental science students and faculties, they are already researchers, they are already aware of this, as it is found to be almost 3 times that of France. So you can just imagine the size of this particular garbage patch that I am talking about. Then once we talk about 80% of all marine debris found in the oceans, they are made up of plastics. Okay, and it is being estimated that 11 billion coral reefs have got 11 billion microplastics are entangled on these coral reefs. And as per the estimate, approximately 51 trillion microplastics as well as 25 trillion macroplastics are found in the ocean water bodies. And it has been speculated that 100% of baby sea turtles, they will be having plastic in their stomachs. And nearby future, it has been estimated that the number of microplastics in water bodies, it will be larger than number of fishes present in the ecosystem, aquatic ecosystem. You can see some of the pictorial representation of how it can impact the animal life as well, because these harms animals through entanglement, by ingestion, as most of the time they mistake it for food. And not only that, they release certain chemicals in manufacture of plastic. Also during its manufacture, various additives are being added, like phthalates and other additives are there, which are very much harmful, and they cause various toxic effects. And these toxicity can lead to injuries, it can lead to starvation, and ultimately leading to death. And this not only is leading to death of the biological species, it hampers the complete ecosystem. This is a slide with which you can have an idea how this microplastic is impacting human health also. They can enter microplastics and nanoparticles through food, beverages, etc. It can be ingested by human body, and it can enter the gastrointestinal tract, affecting the liver. Similarly, it can be inhaled from the air also, and thus it leads to through lungs, it can lead to blood and lymph. And then it has been also observed that these microplastics, they are not only found in the reproductive organs, placenta, embryo, kidney, but they are also found to cross this blood-brain barrier, and they are also being observed in brain. And this is a serious matter of concern that we have to take care, because these microplastics are nowadays found everywhere. Not only they are found in the food that we are eating, in sugar, salt, and in every aspect of life. Due to excessive uses of these plastics, it has been observed that they are found everywhere. And it has been observed that even in the tea bags that we are using, which is most of the time, if it is made of polypropylene, they are also having large amount of microplastics being released. And not only microplastic, even nanoparticles are also being released. And it has been observed that those tea bags which are made up of polypropylene, around 1.2 billion these microplastics are observed per milliliter. So you can just have an idea how much amount of microplastics are being released. And the tea bags that we are using, we have to take care that it should be made up of natural materials. There should be no, even the paper cups that we use, that is also having good amount of microplastics to provide strength, or to provide a particular shape, as you can say. For the tea bags, a pyramidal structure is required, and there we add microplastics. So thus there are various potential health risks related to these microplastics. The health risk includes inflammation, endocrine disruption is there, and definitely possible carcinogenic effects could arise due to absorption of these microplastics. Then there is great requirement of toxicological studies to unveil how these microplastics are interacting with the biological systems. And we have to evaluate the ingestion pathways as well as the systematic implications for human health. There is requirement of regular regulation on this allowable level of microplastics, especially in food and consumer products, to safeguard the public health. Then the social and economic impact, apart from the human health, animal health, and aquatic health, they have got a social and economic impact as well. They affect the communities, especially those who are reliant on tourism and natural resources. It affects on local economy, definitely those which rely, it will lead to decline in the tourism and fishing industries, and thus economical hardships could be there. Health concern for local population, those which are exposed to high levels of plastic, they also faces large number of health issues, and definitely there is a huge economical cost involved with the cleanup process. Now, talking about the sampling part of this microplastic and its characterization, basically we use samples from sample it from biological samples, wastewater samples, water samples, as well as sediment samples. After sampling, separation methods are being used, and initially the most common separation method includes density separation with the help of flotation, centrifugation, and ultrasound sonic separation. Followed by that, we go for digestion with the help of enzymes. Enzymatic digestion is being carried out. And then we have filtration mechanism also by mechanical filtration using filters, sieve plates, etc. That is, filtration is being done. Followed by that, there are certain analysis methods are being involved for this microplastic analysis. We used microscopes. SEMTEM is also being used. We also use Raman spectroscopy, FTIR. Nowadays, this thermal desorption, GCMS, is also being used. Pyrolysis, gas chromatography, mass spectrometry is also being used to identify the chemical nature of the polymers which are present over there. These are some of the methods by which separation is being done. Like with the help of enzymatic action, we do separate this. There are certain electrostatic separation methods also. Density separation as well as magnetic separation is also there. Some of the remediation methods which are most commonly used related to these microplastics, it includes membrane filtration method, advanced oxidation processes with the help of adsorbent, which is now a thrust area of research, bioremediation, phytoremediation, we use coagulation, flocculation, and sedimentation as well. And this adsorbent are emerging as a thrust area of research, especially in the microplastic removal. You can see various adsorbent has been used during the recent years, like sponge activated carbon, carbon nanotubes, aerogels, biochars, as well as layer hydroxides. And this is just a representation of the number of publications which has been found associated with removal of microplastic by adsorption at Web of Science. It has been found increasing since 2020. And just, I would like to just go fast. The microplastic removal using biochar, that has been done in recently, and this is also one of the research area that we are also interested. It has been observed that when we pass this microplastic through the biochar column, it removed up to 92% of the microplastic. And we have used biochar, which is made up of pine wood as well as sugarcane. And certain definitely future research is required, which needs to scale up this biochar based remediation techniques, and we can integrate them into agricultural practices to reduce the microplastic contamination effectively. This is some of the research part which is important related to microplastics. Then the sustainable solution, as all of are aware, this 3R practice: reducing, reusing, recycle. Definitely, we need to have, before switching off to another products which are not made up of, or completely banning this plastic, we need to have reuse alternatives for that. So definitely, first thing is that we have to look out for alternative solutions of all those plastic products, and we should go for innovative packaging solutions also, which are basically biodegradable in nature. So that is also a thrust area of research. Some of the global efforts which are required, there are various international agreements to reduce the plastic production and improve its waste management practices. Regional bans on single-use plastic is very much required, and definitely the corporate sector could also play a very important role by taking off the responsibility to provide sustainable practices in terms of this plastic uses. Now, coming to the individual level, definitely until unless an individual is not involved in any activity, no success could be achieved. So we have to take individual actions. We should adopt mindful consumer behaviors, which contributes to a more sustainable plastic economy. And wherever required, we should definitely discard single-use plastics, and we should go for those projects which are sustainable in nature. We should choose sustainable products like reducing reliance on single-use plastic and encouraging market demand for sustainable action. We do require community activities also, community action like awareness at community level, taking up the community for cleanup drives, and thus there will be a sense of belongingness and ownership will be also developed, and that will be responsible for resilience or improvement of the local ecosystem. Then, as per the time schedule given to me, I just come to the concluding point that whatever we have discussed, we tried to explore the critical aspects of this plastic pollution, the impacts that it is having, and the potential solution that require immediate attention and action to be taken. Addressing plastic pollution is vital not only for the ecological integrity, but also for the health as well as sustainability of communities throughout the globe. And there is an appeal to all the audience, whomsoever is listening. Not only they have to be sensitized, they have to sensitize others also to adopt sustainable practices, participating in advocacy and spread awareness within the community so that this plastic pollution could be mitigated, or at least it can be minimized. And together, through consistent efforts and collaborative strategies, we can effect meaningful changes and strive for a future free from plastic pollution. So I hope I tried to finish this presentation within the time duration whichever was being provided to me, and I tried to give justice to the opportunity which is being provided to me. Thank you very much.
Thank you, sir. Thank you, Dr. Sir, to complete your lecture timeline. And waiting for some questions to ask questions.
If there is any query or any question is there, definitely.
Hello, sir.
Hello, ma'am.
Hello, sir. It was a very nice and informative presentation. Sir, just I want to know that you have discussed many of the remediation techniques for the microplastic.
Yes,
so I want to know that which of the technique is highly effective for the management of microplastic.
Ma'am, simply saying, none of the approach is 100% sufficient enough for dealing with these microplastics. But as per the research which has been observed during the last 5 to 6 years, you can say, this biochar has emerged as one of the most effective adsorbent for this plastic remediation. So currently we are trying to just focus on this biochar so that good amount of results, because very good results have been found to be obtained from them. More than 90% adsorption of this biochar is there, and definitely it is eco-friendly in nature. It is also not having any harmful effect or the side effects associated with them. So right now, so I can say this adsorption, especially this biochar one, is most effective.
Okay, sir. Thank you so much.
Thank you.
So, many types of lectures are really microplastic is the reason for world conservation. We have to involve in the project by research. Thank you, sir, for lecture. So now we are waiting for Dr. Santosh Paul ma'am. So hope you are here. So just before the calling, I would like to introduce Dr. Santosh Paul ma'am. He is a senior assistant professor at the Department of Environmental Science Engineering, Science and Engineering through Jambleswar University of Science Technology, Gujarat, Haryana. Dr. Vishal completed his doctoral research from Punjab University, Chandigarh. He has published more than 6 articles with national and international journals. Dr. Vishal is a recipient of the Young Scientist Award from the Baby Ram University, 2012. He was Dr. Vishal is a recipient of the Young Scientist Award from Baby Ram University, in 2012. He was awarded for defense by India. We have a wide range of nanotechnology, college of management, and research treatment. We have significantly contributed to the field of nanotechnology and wastewater treatment. We have given a national international lecture in almost more than 40 conferences. We are working on a project on nanotechnology for wastewater treatment that we see by using from I would like to give Dr. Salma to
Thank you, sir. Thank you for a nice introduction. And I thank you the organizing committee for inviting me for a presentation. So can I share my presentation? It's visible, sir?
Yes, it is okay. Okay. Okay, it is visible.
It's visible. Very good. So can I start?
Yeah.
Okay, a very good morning to one and all present here. I'm Dr. Santosh from the VIKSIT University of Science and Technology, Gujarat. As we all know, the VIKSIT Bharat ambitions is become a developing nation by 2047 and achieving zero emission by 2070. The idea of VIKSIT Bharat is basically the world-class infrastructure, poverty elimination, and social equality, and industrial strength, innovation, and climate resilience. Various measures has been taken by the government for the VIKSIT Bharat, like national green hydrogen mission, targeting the 5 million tons of green hydrogen production. There is researcher also work on the green hydrogen production and renewable energy core production initiative and budget by 2027. So my presentation is basically on the innovation of greener nanotechnology for the sustainable future and the VIKSIT Bharat. If we see, the world seek a very sustainable solution from the environmental challenges. In this, the nanotechnology is out of as one of the powerful tools which have immense potential. Green nanotechnology have integrated environment and human health consideration into design, production, and application of the nanomaterials. Particularly, this greener nanotechnology is emphasis on the eco-friendly method, resource efficiency, minimum toxicity, and benefit to the nanoscale innovation while reducing the ecological footprint. This nanotechnology, greener basically, sustainable shift not only for the greener production but also pave a way for a safer, more responsible and scientific process. If we're looking at the history of the nanotechnology, nanotechnology is the matter at the atomic and molecular scale, first conceptualized by the Richard Feynman in 1959. Plenty of whom is available at the bottom. But the nanotechnology basically focus on the material science, electronic, data storage, medicine, drug delivery, nano coating, and semiconductor. But by the early 2000, they began rise potential toward the toxicity of engineering nanoparticle and their unknown effect on the on the ecosystem. Thus, they give the awareness to the birth of this particular greener nanotechnology by the principle of greener chemistry and sustainable engineering for the nanoscience. Greener nanotechnology play a critical role in the cleaner energy, water purification, and antimicrobial property, metal sensing, biodegradable nanocomposites. Basically, if application trend of this particular greener nanotechnology, the data covered on 2010 to 2014, there are but slight peak higher in the 2012 and 2014 onward. There are 40 research publication by 2018, and if you move on 2019 to 2014, there are number of publications. 81 publication is published in 2003 on the greener nanotechnology particle. The basic principle behind this particular nano technology, how we can say this particular nanoparticle is greener. There are the several principles which working on the greener chemistry and greener engineer for the design, production, publication, which ensure basically the environment and the human safety. First one is the design of environment green product, which is non-toxic and non-bioaccumulative and easily recycled, and use of this safer material, energy efficient, means minimum energy consumption at the room temperature and pressure condition, and use of renewable energy resources like plant-based materials, petroleum-based material basically use the synthesis of this particular nanoparticle, and reduce waste, safe life cycle design, and real-time monitoring, minimize accidental and harmful byproduct of this nanomaterial, and minimum exposure and risk, and easily recyclable and reusable material, and green solvent and can. If we talking about the various synthesis of greener nanoparticles, there are different approaches for the synthesis of greener nanoparticles. Basically focus on the hazardous use of reduce the hazardous chemical, minimize energy consumption, employing renewable and biocompatible resources. You can use plant-based material like plant extract, leaf, fruit, root. We produce a nanoparticle from this, and we can use microbial synthesis, bacteria, fungi, and algae. And this is the common synthesis material which used using the biological resources, the literature available on this particular nanoparticle, synthesis from the biological resources. We can use the different material for the greener synthesis of this particular nanoparticle. In our lab, we synthesize material from the waste also, eggshell. From the eggshell waste, we prepare the calcium oxide nanoparticle, and water barrier one of the use a biochar, and we prepare a biocomposite from this particular waste, and use their application for the removal of emerging nanopollutants. And we also synthesize nano materials from the plant-based extract. We synthesize from moringa and neem, zinc oxide, copper oxide, and their application in antioxidant, antibacterial, and antifungal. And further, we can use as a nano fertilizer in the field. And here today I present only algal mediated nanoparticle, multifunctional nanocomposite for the sustainable approach toward the environment application. Here we prepare the algal mediated nanoparticle and their application in the different field. This is the common methodology which I use for the synthesis of this particular nanomaterials. And then we characterize using the different techniques like FTIR, one of the basic techniques we use for the characterization of these nanomaterials. We found the functional group in the range of 4 to 600. We confirmed the preparation of this particular nanoparticle. Then the XRD, from XRD, we confirm the phase identification of this particular nanocomposite. Then UV visible for the optical property, Raman for quantitative and quantitative analysis, PAT for surface area, PSA for stability and size, and FSM, VSM for the magnetization, PGA for the thermal stability, and XPS for the elemental composition. First one is the characterization by using the XRD. Here we can see for the XRD, the peak sharp is observed around 18.87, 34.58, which confirm the preparation of this particular nanocomposite, iron, titanium, and graphene nanocomposite. And the particle size is calculated by using a device-Schroeder wave equation found to be in the range of 8.7.42 nanometer. And the vibrating sample magnetometer is used for the magnetic property. Curve is be obtained from the hysteresis loop, which confirm the formation of confirm the magnetic behavior of this particular nanocomposites. We can easily separate this nanoparticle after the reaction. The magnetization is found here is 4.2 to 1 MU 3 per gram. And FESEM, from the FESEM, we can see here the layered structure of graphene sheet and confirm the presence of wrinkled folded graphene layer and hallmark of exoflated graphene. The matrix of this graphene can be assessed and uniform dispersion of all the materials, which indicate the good interaction with the polymer. We also optimize the different parameters for the basically absorption of heavy metal, lead and cadmium. The optimized parameter for the lead and cadmium removal, the pH 6, and dose for the lead and the chromium is 90 and 50 ppm concentration, and nanocomposite dose is 160 and 170 mg per gram for the removal of lead and cadmium. The value of Qmax is found to be 1.6.81 and 30 gram respectively for the lead and cadmium. And R square value for all the cases is found around 1, which indicate it follow all the models, isotherm model for the removal of lead, removal of particularly lead and cadmium. And the antibacterial activity of this nanocomposite, we done the nanobacterial activity with the gram positive and gram negative bacteria for these samples, and we as the concentration is increases from 50 to 1000, the zone of inhibition is increases, which indicated this material is good used for the environmental remediation for the antibacterial property as well. Then the we also use this material for the sensing purpose for the lead and cadmium, and the peak concentration is rising for the cadmium to 5 micrometer, and the cadmium even the low detection even at 0.072 micrometer. Of these particular heavy metals, lead and cadmium. And this particular algal mediated nanocomposite show the significant environment application with challenging the mineral chemistry principle, and they shows the good absorption efficiency for these particular heavy metals, particularly cadmium and lead, and show a good antibacterial activity against the gram positive and gram negative bacteria, and they detect even very low concentration. Means these particular nanoparticles contribute to sustainability, public health, and environment preservation. We can use this particular nanoparticle. We're talking about the role of this greener nanotechnology. It's used in environmental remediations, medicine and healthcare, as we showed in our antimicrobial and antifungal property, we can use this particular nanoparticle. And for the agricultural field, in our lab, we also use this particular green nanomaterials for the nano fertilizer. Check this particular shoot, and we also translocate. We can transfer this particular nanoparticle, and we check the toxicity of this particular nanoparticle in the plant. This particular nanoparticle, and for consumer, these nanoparticles further used in the material, textile, cosmetics, which reduce particularly the toxicity and improve the performance of in particular applications. In the vision of VIKSIT Bharat with a greener nanotechnology, environment sustainability, this particular nanoparticle, I already says, used in different application, air quality improvement and soil remediation, supporting the goal of Swachh Bharat. Second one in the health and sanitation. These nanomaterials enhance the affordability diagnosis, target drug delivery, and antimicrobial treatment. In agriculture field as a nanofertilizers and doubling farming income. And cleaner energy and infrastructure like Ujjwala Yojana and Smart City Mission, Make in India and startup. Greener nanotechnology promote research and development project and eco-friendly manufacturing, Make in India and startup India initiatives. And global leadership in sustainable innovation also. By leading this greener nanotechnology, India can position itself as global hub for the sustainable science and technology and contribute in the international climate and developing goal for the sustainable development goals of India. The future perspective of this greener nanotechnology scale up green synthesis material, we can achievement of biotechnology tool which allow the mass production of this nanomaterial by using eco-friendly techniques and integration with the circular economic model. As we said, the for the preparation of this particular nanoparticle, artificial intelligence and machine learning. With the help of machine learning, which we can optimize the green synthesis processes and predict nanomaterial behavior, improve safety and efficiency. In one of my research paper, I also use the machine learning data for the improved safety and efficiency of this particular nanoparticle. Regulation and standardization for commercial adoption and interdisciplinary innovation for driving novel and sustainable application of this particular greener nanomaterials. This is my research lab, and this is my some research publication in the greener nanotechnology. Thank you so much, sir.
Good afternoon, sir.
Good afternoon.
Ma'am, you have given very nice information regarding nanoparticles. My question is, in your study, which nanoparticle has given lowest limit of detection for the any kind of pollutant?
Actually, this is basically, we can say it's a different particle, different nanoparticle giving a different lower limit of detection for different pollutant. Different nanoparticles have a different limit of detection.
Yes, ma'am. And ma'am, you have explained many characterization techniques for the characterization of nanoparticles. So generally, we are using XRD for the characterization. Apart from this, we can use any kind of technique as well for the nanoparticle characterization.
First one is the FTIR, which gives the basic idea for the confirmation of the formation of nanoparticle. If you found the peak in the scale of 400 to 600 nanomaterial, then we can say metal oxide peak is formed. This is the preliminary characterization. But from the XRD, we confirm basically the formation of that particular nanoparticles. After that, we can use the different technique for the characterization of different purpose. Like I'm talking about the SEM. This is for the surface modification, but the confirmation is from the XRD or from the FTI. The major confirmation is only from the XRD itself.
Okay. Thank you, ma'am. Thank you so much.
Thank you, ma'am. So now,
Actually, sir, you are not audible.
So
Yeah, now it's now you are audible.
natural and artificial intelligence impacting quality ecosystem under climate change. Saurabh Pandey. So Saurabh Pandey is there.
Sir, can you please type here? Actually, your voice is not clear. Yeah.
Is it clear now?
Yeah, sir. Now it's clear.
Okay. Thank you, ma'am, for informing us. So CP76, natural and anthropogenic sedimentation impacting aquatic ecosystem under climate change. So Saurabh Pandey is here. Saurabh Pandey. Okay. Next, CP77, sustainable technologies for VIKSIT Bharat. Chitra Kumar. CP78.
May I stop the audio?
Just I'm taking the name. If present, please tell me. Okay. So CP78, sustainable dyeing technique, advancing eco-friendly textile processing for VIKSIT Bharat. Madhvi Tomar. Next, transforming historic structure, the role of adaptive reuse, modern sustainable design. Manish Kumar Chitransi. Okay, next. Manu Chandel. Okay, next. Bhavna Sikarwar, nanotechnology and its application in photovoltaics. Next. Chitosan encapsulated anti-larval essential oil nanodispersion for the Anopheles mosquito control. Vijay Lakshmi Ghosh. Dr. Vijay Lakshmi Ghosh here. No. Fabrication and characterization of the biocomposite film based on Gurugram, chitosan, polyvinyl alcohol, tannic acid, and silver NPS for food packaging. Pallavi Mandil is here. Pallavi Mandil. Not getting. Next, Aditya Bhattacharya. Sustainable architecture, material, and nanotechnologies, promoting eco-friendly building practices. Okay. Next, Aditya Singh Baga. Gurujot Singh. Impact of Swachh Bharat Abhiyan on school sanitation, a review. Next, advances in photocatalysts using tin sulfide nanoparticles, sustainable solution to dye removal. Kajal Bist is here. So need to remove. Actually, few of the present CP already presented. Next, diabetic retinopathy detection using the deep learning. Bibhav Sikarwar.
Yes, sir, I'm here. Bibhav Srivastav.
Okay. Yeah, you can start.
Yes, okay, just a minute.
Next, Madhurima, and followed by Manish Kumar. Okay, sir, you can start. It is visible, sir. It is visible.
All right. Good afternoon, one and all available in the meeting. I, Vibhav Shankar Srivastava, Research Scholar Assets, Amity University, Madhya Pradesh, Gwalior. And today I'm going to present my presentation over the topic which is related to the disease. This is diabetic retinopathy detection using deep learning. Now, let's discuss and understand what is diabetic retinopathy. Diabetic retinopathy is a disease which happens to the person who suffered with the diabetes, and the cause of this disease is damage in the blood vessels of retina. So there is a four stages are available: mild, moderate, severe, and proliferative disease, retinopathy. In a different stages, different type of images are available in this PPT. You can see the first stage of this image, second, third, and fourth stage. This is the normal. So what our work is actually, as far as concern of the research work, we have gone through a lot of research papers, and the work has been done into this area. A lot of people done CNN using the CNN network and provide the framework, segmentations, methodology to generate the result in a very excellent and accuracy levels. After the reading all available and the suitable research papers, we found a lot of scholarly articles work are available in this field, and there is a still possibility to work on it. So we did some different type of work, because we see most of the work available in this field is only a specific stage, but our work is fully designed for the entire stages in a journal pattern. We find out the disease in all stages, whether the person is really suffered with the disease and what kind of stages he or she undergone. And the methodology we were used to solution of this problem, like firstly we is going with the pre-processing of the data which is available on we train our model from the Kaggle data and extract the feature imaging, and different stages we collaboratively done and design an algorithm to isolate these stages and validate this algorithm. After the validation, we can go for the implementation. So this is our work, and the outcome after our work is like there is no tool available in the market specifically to identify this disease and provide a journal outcome, journal result, so that patient can detect their disease initial level and can be managed with the medicines. Thank you, sir.
Okay. So, Vibhavji.
Yes, sir.
So, how we can recognize whether someone have it means some diabetic retinopathy, it means diseases. So how can we rectify?
All right, sir. Sir, we can find out, first of all, prerequisite is the person or the patient must be suffered with the diabetes. The person who suffered with the diabetes, he can be, the possibility of diabetic retinopathy more than it. And there is a diagnostic process which is called fundus imaging. Fundus, to analyze the fundus imaging and the images analyzing, extracting the imaging and segmentation of the imaging, we can identify, or our model identify automatically this person is suffered with disease or not. So by the use of fundus imaging, we can isolate the person who suffered with the disease or not suffered with disease.
What I mean, some background data is already available to match all that pictures.
We use two type of data. One is data taking from the Kaggle data set for training our model, and another real-time data which is available by collaboration, Ratan Jyoti Hospital, locally available in Gwalior.
Okay, thank you. So anyone? Okay. So Madhurima.
Thank you, sir.
Madhurima ji, are you available here?
Yes, sir, I'm available.
Okay.
Am I
So
audible?
yeah, you
Could
are
I leave,
audible.
sir?
You can start your presentation.
Okay, sir.
Thank you, sir.
Sir, is my screen visible?
Still not, but I think you are doing sharing.
Is it visible now?
No, it is not visible. Share screen, please.
Yeah, I have shared screen, sir.
Yeah, wait. One second.
Sir, some allowed option they are showing.
शेयर करने का ऑप्शन आ रहा होगा इन पर।
आ रहा है, मैं कर रही हूँ मगर वो दिखा नहीं रहा है।
शेयर का ऑप्शन आ रहा है आपको?
या आ रहा है सर।
If she is able to unmute ना, she can share.
Sir, can you hear me?
Yeah.
You are audible. You are clearly audible. Please upload your PPT.
Yeah, I'm uploading, but it is not showing in the screen. I don't know why.
You are from mobile.
Yes, sir.
So then share your screen through this. Don't share the window. Just click on the share button and share the screen only. Don't click the window.
No, sir. I've done the screen only. Share screen.
Just
I'm doing.
then minimize this and open your PPT. Minimize it and open the PPT.
Sir, is it visible now? I've opened the PPT.
It's not visible.
First you do screen share and click the share.
Better you use the laptop.
Yeah, I have done that only. I'm clicking share and then only I'm putting the PPT and share screen only I'm doing.
After share screen, what you able to see?
After share screen, it is showing that allow Zoom to record or cast your screen. I did it start now.
You just allow it.
Yeah, I've allowed. This permission allows an app to appear.
Arvind ji, please check this issue. Arvind ji.
Is Arvind ji able to hear us?
अभिषेक सर, आप तब तक नेक्स्ट कराइए, इनका बात करके अरविंद जी से करते हैं शॉर्ट आउट।
Okay. So, ma'am, please.
Meanwhile, ma'am, you do one thing. Just leave the meeting and again join.
Yeah. Okay. So I'm calling to next CP89. Devendra Kumar, assessment of water quality Hindon River at Saharanpur. Next, CP90, drone taxes, drone deliver in India, examining the legal and ethical dimensions. Nidhi Kastwar, if audible. So CP91, CP92, CP93, Charu Sharma, food security and sustainability. CP94, Niharika Singh, enhancing antimicrobial activity and preventing biofilm infections with the plant-assisted nanoparticles. CP95, Ashish Sharma, physical accessibility audit of public building and spaces. Assessment of a smart city, Gwalior. Next, Harsh Raj, comparative analysis of CNN and RNN techniques for ECG. Next, reclaiming domestic wastewater as liquid gold and environmental study on its journey from a sink to soil. Ansu Dwivedi. So, Dr. Madhima, you can try once again.
Yes, sir, I'm trying. Just a minute. Huh?
Okay.
I have rejoined again. I don't know. They are showing some document problem. I think so. Now it will be visible. If I'm not, I'm checking once.
Yeah, please
Just
check.
a minute, huh? Just a minute. It is showing unsupported file. Huh? How it can show? I'm sorry for the inconvenience which is caused due to me.
No issue.
I don't know. I have already done 1,000 times before from here. I don't know today what's the problem is being.
So if not possible, you can send to me and I will share here.
How can I send you, sir?
On WhatsApp, you can send or
Dr.
mail,
AK
whatever.
Dr. AK Bharadwaj, sir, right?
Yeah, yeah. Just I'm writing in it means chat box my mobile number.
Sir, I think so. This is the best option. Thank you, sir. Sir, I've sent you. You can just see it once. Is it visible for you?
Oh, wait, please.
I've sent it in the WhatsApp.
Yeah, yeah. Chat box कहाँ गया?
Sorry.
So hope you received the message.
Yes, sir. I've already WhatsApped you. Can you please see? I've sent it in the WhatsApp.
Wait.
Thank you, sir.
You can start, madam.
Yeah. Thank you, sir, very much for your assistance.
Okay, most welcome.
Very good afternoon. Today, I am here in the International Conference on Sustainable Technologies for VIKSIT Bharat from MIT University. Myself, Dr. Madhurima Juardar, Assistant Professor from SRM IST Ramapuram, Chennai. I'm going to present today our topic on appraisal of acute and chronic arsenic exposure on school children from exposed and appropriately controlled areas of West Bengal, India. So the next slide. Yeah, so the first highlighted point is the groundwater arsenic contamination scenario in West Bengal. As we know that India is the largest user of groundwater in the world, more than 60% water percentage is used for agricultural purposes and 85% for drinking purposes. So as we all know that arsenic contamination in drinking water has been a recurring problem in today's world, but that private tubewells are more than 90% of the existing hand tubewells. So this is a publication which has already been published in 2010. Followed by the next slide, where we find that apart from consumption of arsenic contaminated drinking water, for a long period of time, contamination through foodstuffs are equally responsible for causing several health hazards. Next slide, sir. Now, we can see that not only drinking water, this arsenic entry in the food chain causes through the agricultural shallow groundwater through cultivation practices in the whole paddy plant through the rice, it gets translocated to the rice grain. How? Through the root, then stem, then leaf, then the whole grain, and then the rice husk, and apparently to the rice grain which we consume in our daily life. Followed by the next slide, where we have the study area locations. Sorry. Sir, the next slide, please. We have two locations where we have taken apparently control areas, Kolkata and exposed area for Gaighata. Now, this Gaighata is totally arsenic exposed area, followed by Kolkata, which is the apparently control area. Next slide, where we find that the assessment of acute and chronic exposure of arsenic and future risk of suffering have been investigated on school children from highly exposed Gaighata area and apparently controlled Kolkata area of West Bengal, India, where we have certain objectives where we find that the cumulative exposure of arsenic toxicity through the consumption of drinking water and rice grain, qualitative analysis of arsenic in the biomarkers of arsenic, which is urine, scalp, hair, and nail, evaluation of future health risk, assessment for school children, follow-up health exposure after 8-month study to put forward the outcome of awareness program on arsenic toxicity. Followed by the next slide, where we find that the exposure of arsenic toxicity through daily dietary intake, mainly through drinking water, and arsenic contamination through the grain, like the rice grain and the vegetables, is transported to the exposed and apparently controlled children, where we find that our study reveals a potential area of concern related to subchronic exposure in children due to arsenic toxicity through drinking water and rice grain. Followed by the next slide, where we find that is rice grain safe apart from drinking water? So the next slide, please. Yes. So the daily dietary intake of arsenic through drinking water, rice, and midday meal for the school children from exposed area is higher compared to the apparently controlled children. Now we can see that the parboiled rice grain, that is a daily consumption rice grain, is transported highly in the exposed children, followed by the apparently controlled children. The daily dietary intake rate of arsenic through drinking water for the exposed children is much higher compared to WHO recommended value, that is 3 microgram kg per body weight per day. Followed by the next slide, where we find that the health risk exposure is highly accumulation of arsenic has been observed in biomarkers of urine, hair, and nail for the exposed children compared to apparently controlled children. Now here we find that the risk assessment for the school children for the studied population through drinking water and rice grain, we find that the exposed area children are exposed highly compared to the apparently controlled area through drinking water. But in the case through rice consumption, the distribution of cancer risk is equally distributed, nearly 45% and 55%, because the rice grain which we are consuming in both the controlled area or apparently controlled area and the exposed area is being transported from our mainly arsenic contaminated zone to the population of other location which is not actually arsenic contaminated. So the cancer risk assessment is high through the consumption of arsenic contaminated drinking water for the exposed children compared to the apparently controlled children. Intake of inorganic arsenic is considered as a potential area of concern depending on human health hazard. Contribution of inorganic arsenic is high, approximately 89.5% through the consumption of contaminated rice grains which are being cultivated in exposed area and being transported to the control areas. The probability of cancer risk is almost equally distributed through ingestion of contaminated rice grains for both the studied air populations. followed by where we find a health exposure study was conducted after 8 months on the exposed children, and the study highlights that the significant decrease in arsenic next slide, sir. Yes, yes, sir, that slide only. A follow-up health exposure study was conducted after 8 months on the exposed school children. The survey highlights a significant decrease in arsenic concentration of urine and other biological tissues of exposed children after minimizing the level of arsenic contamination. This shows an awareness program on severity of arsenic toxicity and supplementation of arsenic-free drinking water from a nearby organization are main sources of a noteworthy declining trend of arsenic accumulation in exposed children. Summarizing the whole study, which has been already published in our Health Exposure and Health Journal, here we find that the children are subclinically affected due to arsenic toxicity. The daily dietary intake of arsenic or exposed children is highly for the exposed area children compared to the apparently controlled children. Higher accumulation has been observed in the biomarkers for exposed children. Cancer risk assessment is high through contaminated drinking water, but contribution of inorganic arsenic is almost equal through the consumption of contaminated rice grain for both the studied areas. So overall, from the summarizing study, we can suggest that the awareness program on severity of arsenic toxicity is required on a daily basis till today, and supplementation of arsenic safe water for a noteworthy declining trend of arsenic accumulation in human body system, followed by the mitigation option. Not only that we are having the accumulation or the effect of the arsenic in the today's world, we followed in the next slide, we show that the mitigation option is highly required, that is availability of arsenic safe drinking water for a sustainable environment. Using surface water or rainwater is obviously for the best choice of cultivation process so that the accumulation rice grain doesn't occur in a frequent way. In this case, we can see that the apparently controlled area and the control areas are also arsenic free because rice grain or the transported vegetables are accumulating arsenic in today's world. Minimizing the use of arsenic contaminated groundwater for agricultural practices to reduce arsenic accumulation in paddy as well as seasonal vegetables, which will lead to a sustainable society. Followed by the next slide, where we find that overall concluding that arsenic exposure is not only the highlighting study in today's world, where arsenic is being transported through water, dietary foodstuff, rice grain. There are severe health effects, that is non-carcinogenic as well as carcinogenic and skin lesions. The main part should be highlighted as the intervention options or the mitigation options or the remedial strategies, where we find out that the safe drinking water, food, proper nutrition, proper management, proper mitigation policies, irrigation and cultivation practices, community participation, awareness programs must take place to have a sustainable development society in today's world and a very safe place for the people of rural Bengal to stay in a healthy life. So thank you, and be aware of the arsenic pollution in today's world. Thank you very much. And thank you, sir, for helping me presenting the slide today in the conference.
Oh, most welcome. So you presented your things very nicely. So we have some question regarding it. So you as told us, it means whatever you explained. So is it arsenic contamination possible by the food stuff? It can be transferred if we grow in that area.
Yes,
so,
sir.
1 second. So, sorry for, yeah. So what I'm asking, so if we produce rice or whatever food stuff in that agriculture area and people sell outside of our country, so it may transfer the arsenic toxicity in other place also, maybe?
Yes, sir, because where we are cultivating that agricultural soil and the water which we are using for cultivation, that is the shallow water, if that area is arsenic contaminated, it is being transported to the rice grain as well as the seasonal vegetables. And mainly we know that the cultivation process takes place in rural Bengal. So every food we are getting is transported from rural Bengal to the urban areas. In this way, the arsenic is getting transported and we are slowly taking the poison inside in day by day. Slowly it's going, sir.
Okay. So on which group of age group of the school children having a more prone to that arsenic poisoning? Have
Sir,
any
it's
data?
as we know, arsenic is a slow poison, sir. So this study was done for young children with their growing stage. Mainly when they are consuming on a certain till 12 years, if they continuously consume, no, so they will start showing the dermatological manifestations, which we have seen also in the study areas. We have
Okay.
gone for field research and we have seen that when they are consuming it for last 12 years, we know it's a slow poison, it is a long term, we have the chronic effect. In acute exposure through urine, we can see mainly the acute exposure through the children. In hair nail, they are having deposition, but slowly. They'll have the deposition in the early age. But when it is after 12 years, when they are growing 13 years, 14 years, slowly they are growing. So that is visible. Slowly they will be having the skin lesions, their arsenic accumulation in the hair and the nail will increase. In this way, we are having the datas.
Okay. It means physiological data you recorded.
Yes, sir. We have the surveyed data also, and we have also measured it in the double AS,
It means
atomic
whatever
absorption
you
spectrum.
explained here, that is that it means research work or totally review work?
No, sir. It's a totally research work. It's my research work. It's my PhD research work, which I feel
Okay.
it's my. I really felt very honored by doing this particular work because I have interacted with the children of that age, and really, sir, it's very much pathetic to see their situation. Their parents are suffering a lot, so we don't want to know that the children when they grow up,
Okay.
they go in that same stage. So I'm very honored that I could work on this and I could present this topic today in
Okay.
this.
Thank you. Thank you very
Thank you,
much,
sir.
Dr.
Thank you
Madhurima.
very much.
Have any question you can ask participants. And so I took the name of all candidates who listed here in this session. So if anyone present, please raise the hand and present your topic, whatever you have. So anyone who fall the technical session, whatever 5, anyone present? Okay, so nobody here. So if you're not responding, so I will close here. So please respond if you anyone here. Okay, so now one thing before the ending this meeting, thank you to Professor Asutosh sir and Dr. Bhupal ma'am for your presence. And the second thing, I request to all participants to join again us at 3 o'clock. It means 3 p.m. So we are waiting for the your presence again here in a valedictory session. So the valedictory session will start on a sharp 3 p.m. So please join us. So now we are taking a break. So thank you, everyone. We are waiting for you. Hello, Professor Lucien, ma'am.
Hello, Professor. How are
Good
you?
morning. Good morning.
Good morning. Good afternoon for you.
In your country, morning.
Yeah.
Yeah. So
Here
you
it's
take
word.
a little sleep today, I think.
Yeah, a little bit.
That's great.
Can I test my presentation?
Yeah. Sorry. What you mean?
Yeah, I can put here only to test.
Yeah, you can wait. Before that, I think.
Okay,
Yeah, you
it's
can put here.
working.
You can try
It's
again.
working. It's working. Okay. Stop sharing.
Yeah. Your PPT was visible. No issue.
Am I audible, sir? Okay.
Good afternoon, madam.
Good morning for me. Good
Yeah, it was
afternoon
good
for
morning
you.
there.
Hello.
It's
How
late
are you?
afternoon here. It's around 3 p.m.
Yeah.
Yeah. So we are very glad to have you. And
Thank you.
we will start this session in just a few minutes from now.
Okay.
So it must be around 8 in the morning.
Here, 6:30 in the morning.
Okay, it's early morning, right? Right.
Yeah.
Right.
मेरे लैपटॉप का साउंड क्या चला गया?
Arvind ji? Arvind?
And it's a very good coincidence that we have two professors from Brazil itself.
I saw.
Yeah.
Miss Luciane.
Yeah.
Hello, Professor.
How are you, Luciane?
I'm fine.
Very nice. Thank you for the invitation.
Welcome, sir. Welcome.
It's our pleasure, sir, to have you.
Very nice.
Right. Dr. Sabnal, can you make a call to the Honorable Pro Vice Chancellor so that he can. Okay. Let me just brief you, Dr. Ciro and Luciana. We had this conference, a two-day conference. And yesterday, we had the Municipal Commissioner of Gwalior as the chief guest, and Professor Arvind Kumar, who was the former Vice Chancellor of the Central Agriculture University. And he has been awarded the fourth highest civilian award of our country, Padma Shri, for his exemplary work in science and technology, particularly in the development of agroforestry systems in India. And he was our guest of honor. And then we had one session which was offline, and we had four eminent professors from different parts of India, eminent professors and scientists. And in total, we had 6 sessions, of which there were 8 plenary lectures, and around more than 50 contributive papers were presented orally as well as in poster sessions online. So this was a mixed conference, hybrid mode. The inaugural and one session we had in the offline mode, and the rest of the sessions we had in the online mode. And since it would have been great had we been able to have you in this university in person, both of you eminent scholars and professors. And next time we will plan something in such a way that we are able to have professors like you in the campus itself.
Nice. It will be great.
Right.
It's a...
Anvesha, I think we can start now.
Yes, sir. Am I audible?
Yes,
Anvesha, you are audible.
Arvind ji, please make Professor sir and co-host, MP Kaushik sir.
No issue. I am always visible.
Okay, sir. Okay.
All right. Very pleasant afternoon to everyone. It gives me immense pleasure to welcome you all to the valedictory session of the International Conference on Sustainable Technologies for VIKSIT Bharat 2025. We extend our cordial felicitations to the Honorable Founder President, Amity Education Group, Dr. Ashok Chauhan, and Honorable Chancellor, Amity University, Madhya Pradesh, Dr. Asim Chauhan. We extend our heartfelt greetings to all the esteemed dignitaries who have joined us online. We're extremely honored by the presence of our esteemed guest of honor, Professor Dr. Luciana Maria Cola, University of Passo Fundo, Brazil. We are honored to extend our cordial felicitation to our esteemed guest, Professor Dr. Ciro Abodrigi, ASALQ, USP Forest Sciences Department, Piracicaba, Brazil. We are privileged to extend our cordial felicitation to Lieutenant General V.K. Sharma, Ativist Seva Medal, Honorable Pro-Chancellor and Senior Vice President, Vidman Balved Education Foundation, Professor Dr. R.S. Tomar, Honorable Vice Chancellor, Amity University, Madhya Pradesh, Professor Dr. M.P. Kaushik, Pro-Vice Chancellor, Research, Amity University, Madhya Pradesh. To all the deans, directors, head of institutions, head of departments, faculty members, research scholars, and participants who have joined us online. This conference, organized by the Department of Environmental Science, Amity School of Life Science, Amity University, Madhya Pradesh, brought together eminent scholars, researchers, students, and professionals from diverse fields, united by a single vision, sustainability of VIKSIT Bharat. Over the past 2 days, we have witnessed a vibrant exchange of ideas, in-depth discussions, innovative presentations, and inspiring keynote addresses. The sessions were not just enlightening, but also a testimony to the passion and commitment to our academic and research community toward building a sustainable and developed India. We're honored to welcome Professor Dr. Lucian Mariacola, an esteemed academic researcher with deep expertise in food science and engineering. A graduate in food engineering and a PhD holder in the same field, she has built a remarkable career in advanced biotechnological research. Her work primarily focuses on the cultivation of microalgae for bioproducts and environmental applications. She also explores submerged and solid-state fermentation techniques, contributing significantly to sustainable environmental technologies. Professor Kola has been a dedicated faculty member at the University of Pacific since 2002, actively guiding master's and doctoral scholars in the field of civil and environmental engineering, food science and technology, and agronomy. MAM's academic presence spans across multiple undergraduate disciplines, including food engineering, chemical engineering, agronomy, and pharmacy. Please join me in extending a warm welcome to our distinguished guest, Professor Dr. Luciana Mariacola. Thank you, ma'am, for joining us today. You're kindly requested to deliver your address. Ma'am, please.
Thank you very much. I will share my presentation. It was an honor to be here with all of you today. Are you seeing?
Yeah, it's visible, ma'am.
All right. Yes, ma'am. Okay. So, good afternoon for everyone. I will present my speech named Microalgae as a Platform for Climate Change Mitigation and Food Security. I am a professor at the University of Passo Fundo. The University of Passo Fundo is located in the south of Brazil, just here. And we are a university that has 6 campi in and we can impact more than 150 municipalities with 10,000 students and 56 years of creation. Here we can see some pictures of our campus when I carry out my activities. So we know that the agriculture can impact the climate change, and that the impacts of climate change about or over the agriculture. So about the impact of agriculture, we have the methane emissions, the greenhouse gas release in fertilizer production, the contamination of soil and water with agrochemicals, the deforestation, and the fuel use because of the transportation of these commodities. In the other side, we know that the agriculture is impacted by extreme temperatures, loss of biodiversity, changes in rainfall patterns, and spread of disease and pests. Why do microalgae constitute a platform for this confrontation? Microalgae are related to several themes, including circular economy and bioeconomy, and making part of the theme of biorefineries. These themes are related to environment with food security, and these are issues normally impacted by climate change. We know that it is estimated that for every kilogram of microalgae biomass produced, about 1.8 kilos of CO2 are fixed. This microalgae can be produced using exhausting gases of industries and nutrients found in effluents. Here we have a graphical abstract that show that this biomass can be produced using sunlight that is a source of energy with very low cost, and the source of carbon can be the CO2, and the other nutrients as nitrogen and phosphorus and other micronutrients can be obtained of wastewater treatment. The bioproducts that can be produced by microalgae could be biofuels, animal feed, biohydrogen, biochar, and other products that can be used in the chain of food and pharmaceutical. Here we can see some works that we did with my students, and here we are presenting the use of very types of effluents to produce biomass. In this one, we used the post-treatment effluent of anaerobic digestion of cattle to produce biomass, and we used here species of spirulina and syndesmus, and in consortia, and after of the optimization of the cultivation, we did the biochemical composition of biomass to make application of this biomass. Here we used the effluent or residues of whey protein concentrate in order to improve the carbohydrate content in microalgae spirulina, with the intent of production of bioethanol too. We used still malting wastewater, still with the aim of production of carbohydrates in spirulina, and in this one, in this paper that we published in Algal Research, we studied whey directly in the cultivation, residuals of whey in the cultivation of spirulina, chlorella, and syndesmus in microscopic cultivation. And here our aim was to study the tolerance level and the scale up of the process. What we learn about the cultivation studies with effluents and with this valorization of effluents. The cultivation studies are necessary for each combination of microalgae and effluent. It is necessary to perform the addition of effluents in small quantities to avoid toxicity. It is possible the reduction in media costs based on organic carbon sources. It's important to perform the LCA, the life cycle analysis of each process, to know better the situation of each one. Here, I'm showing for you a study that we are done for the development of this photobioreactor. This work is being done with a partnership of architecture, and the students involved will study the thermic comfort of our lab, because we have the intention of substitute some windows of our laboratory by these photobioreactors. And this is a theme related to with the climate change. We did some studies with bioremediation of solid and liquid media with microalgae. In both of these papers, we studied the bioremediation of emerging pollutants from pharmaceutical industries. In this first one, we produced biomass in culture media, in normal culture media, and in this one in domestic effluent, and these media were added of emergent pollutants. In this one, we used the inactive biomass of spirulina to perform the bioremediation of soil, contaminated with biodiesel or diesel. So we are showing that microalgae is a platform that can be used for many purposes. In the theme of bioenergy and biorefineries, we performed the production of bioethanol by Spirulina platensis, and the biomass of spirulina was hydrolyzed and fermented to produce bioethanol, and the residuals, fractions of the saccharification process and fermentation process were used to produce biomethane. We show that the direct production of biomethane from the entire biomass was better than using the fractions and producing the bioethanol before. And in this work, in the same theme of biorefineries, we produced the biopeptides using hydrolysis of proteins of spirulina. And after, with the residual fractions, we produced bioethanol. We still developed some foods, and I think that is very important in this context of food security, because we can improve the quality of foods with the addition of microalgae biomass. In these both works, we added biomass of spirulina. In the right, in ice cream, with the intention of reduction of sugar and fat by the addition of inulin, spirulina platensis, or phycocyanin, that was the pigment of spirulina. And it was possible to reduce the quantities of sugar and fat used. And in this one, we made the encapsulating of spirulina in alginate capsules, and after we added this in the pasta with the aim of masking the flavor of microalgae and to protect the antioxidant potential of this biomass. And nowadays we have a new application of biomasses of microalgae that was in agriculture. So we know that microalgae could be important biostimulants for agriculture. This microalgae can improve the nutrient absorption in soil and improve the resistance to abiotic stress, mainly stress of water and salt. And there are results showing the literature the protection against these effects and increase in the resistance of herbicides as well. The effect of chemical composition of microalgae in plants can be obtained because we have amino acids, indole acetic acid, cytokines, nitrogen and phosphorus, and carbohydrates and lipids in these biomasses. And this is related with the detoxification of reactive oxygen species, the maintenance of cellular integrity. This can mitigate the effects, as we say before, the effects of water and salt stress, making possible the root elongation that helps in the development of the plant. Nitrogen and phosphorus molecules can be responsible for the water homeostasis and osmotic regulation, and carbohydrates can oscillate in the rotation of humidity in soil, and the lipids in the modifications of the membrane fluidity in plants. Here we are show some small parts of the thesis of the doctorate of Julia Brown. Julia studied the valorization of agro-industrial waste for the production of microalgal biomass and application in agriculture. So before, she studied the optimization of media with whey, and after we used the biomass to the development of biostimulants and the use in agriculture. And here I'm showing part of her study. Here we produced barley, and we used here is the treatment of the producer, and the treatment with the seed treatment with extract of algae. And in this photo, this and this, we made the substitution of urea as fertilizer by the biomass of spirulina in some quantities. Here is 50%, 100%, 80%, and 40% of substitution. And we can see that the structure of the plant is different. We have more roots when we use microalgae. Here, another part of the work with the cultivation of barley for 82 days. We did some treatments without the use of microalgae extracts, and using in the treatments 1, 2, 3, 4, and 5 foliar application or seed treatment before and after foliar application during the cultivation. And we can see here that the length of root and aerial part was better when we used the treatments with microalgae, and this can be seen too in the effects on dry biomass. Here we are showing photographs of a producer of soybean, and the only difference in the management was the addition of microalgae in 5 grams per hectare during the planting of the soybean. And we can see the difference in the structure of roots and aerial part. And it produced 180 kilos more per hectare in this cultivation, and we observed no end of cycle disease. Here, this was my last slide, my last picture, and I'm showing for you a project that we are beginning just right now. We will do the production of microalgae in 200 liters racioways, and after we will develop with this biomass biostimulants, 2 or 3 types of biostimulants with microalgae. And these biostimulants will be used in the cultivation of strawberry and beans to study the water stress, and wheat and soybean to study the water stress and salt stress. And in parallel, we will perform studies of bioremediation using algal biostimulation and phytoremediation together. This work is beginning now in this month, and with the duration of 2 years. And I want to say thanks for all of you, especially to the Dr. Abhishek Bardwaj. I'm very honored to be here with you today. I want to say thanks to the Laboratory of Biochemical and Bioprocess, my lab, to the University of Passo Fundo, and to Brazilian development agencies. And I hope that we as scientists could be free to do our researches, improving quality of the environment and the food security. Thanks for everyone.
Thank you, ma'am, for such an informative presentation, and we are truly delighted to have you amongst us today. Proceeding further, we are deeply honored to extend a warm welcome to Professor Dr. Ciro Abutriki, an esteemed researcher and academician from the Forest Sciences Department at the Luiz de Queiroz College of Agriculture, Piracicaba, Brazil. With an illustrious career rooted in ecophysiological interactions with agroforestry systems, since the onset of his master's studies in 1998, Professor Righi has significantly contributed to the understanding of plant-environment interactions. His extensive research emphasizes enhancing resource use efficiency, adapting plant species to diverse ecological conditions, and promoting sustainable land use. Notably, his work extends to exploring land use changes, traditional farming systems, and their implications for biodiversity conservation, carbon sequestration, and rural livelihoods. His recent tenure as a visiting professor at the Center for Agroforestry, University of Missouri, USA, further exemplifies his international research engagement and collaborative impact. We are privileged to welcome Professor Ricky to share his rich insights and groundbreaking research with us. We now invite Sir to share his words of wisdom. Sir, please.
Okay, thank you, Ms. Singh. I would like to thank you, Mr. Kuldeep Diveda and Swab Pillay for the nice invitation and all the committee. I will share my presentation here.
Yeah, you can share it, sir.
Yeah. Is it good?
Yeah, still, yeah, it's visible now.
Yeah, nice. So we are talking here for, as I know, people from India like histories. We will start with this wave painting, an old painting made on 1800s in Japan under the great wave of Kanagawa. So the waves are transport of mass and energy. So what we can learn and how we can relate this to agroforestry, land use. Okay, it's not, okay. So I made this title, Developing System Architecture to Improve Land Use System, and how we can use solar radiation in a better way, okay? So Piracicaba, our university is in Southeast Brazil, in the state of São Paulo. It was from 1901. This is the main building. This is engineer. It's a big University of São Paulo is one of the biggest here universities. So I would like to start this with a tale from earthworms that lived in the earth, of course, in the soil. And one day, the family of earthworms were eating their soils, and the youngest started crying and complaining about a blonde hair found in his food. So the father started talking about a story about Benedita. Benedita was a lady that lives in the forest and liked the forest and the animals and everything. But she had a not so good understanding of the nature. So she started making things that it was not really good, but in her opinion was nice. So she find a turtle, but it is from soil turtle, not aquatic turtle. And she takes the turtle and throw in the water, and the turtle dies. So all the book, she's starting like this, misunderstanding everything. And one day she found a snake killing a rat. She has no doubt take a pole and kill the snake and save the rat. And she was very happy and started crying about that. Took the rat in his hand and whip out the tears. So, but unfortunately, the rat was contaminated with a very bad disease, and she dies in the end. And you can see here the family of the rats, very big. The family of the earthworms eating the remainings of Benedita. So what we can learn here, this is a story for very young people, that if you don't understand the nature, this can murder you. No? So we need to understand the nature. And we are facing now big challenge with this disconnection from humans from nature. So we need to understand nature, how to insert in just our practice. As production is a necessity, there are many environmental issues all around the world. So we need to develop more integrated systems. How we can do, based on basic knowledge, so we can make new proposals. So I will start talking about many different things and to join later. So what we have, it's the main source of energy is the sun. We have many fights about energy because without energy, you cannot do anything. So we have an opportunity to optimize this free energy that reaches the earth every day. Okay? So in the other way, we have the problem of time. This is a space and time experiment. Huh? So I look for that, and I found two words for time in ancient Greek. Chronos, that is the one that we are using every day. It's a sequential order, is the time of man. And kairos. Kairos is the qualitative nature. So it's the something special happens, the opportunity moment, when you should do things, not like it like this. The opportunity, you can see and perceive these moments. Oh? So space and time is always related as a sun movement, and the clock here you see the pendulum. But in nature, times can pass differently, like this experiment with beans planted in agroforestry and in monocrop. So you can see the shaded plants, the time passes differently, okay? The time as the time here, the shaded, less energy, the time is low. So in the other hand, so to join everything that to our knowledge, we have to basically understand that the structure, we are the only ones that can make this movement because of the insertions, the two insertions different from all others animals. So we can make different things. And look at that. It's the chameleon. He can make and walk in a twig like that without problem because he has different structure. And you can see this picture from Philippines that there are so many trees structure. So the structure will give the ability to make things. So if you join this, you see here how we can think on system analysis, like agroforest system, land use systems. Huh? So this iceberg model, you can see we are here like the penguin. You see the events, but if you go deeper, you can see the pattern of behavior. What trends are there over time, the system structure, and our mental models. We will try to go deeper, increase this leverage so we can change how we can perceive the world. So to make different systems structure that will give you ability to respond to nature so we can change everything. We are in the deepest part here. So agroforest system, as all you know, is this: you mix forestry and trees with crops and animals in space and time. And this intrigued me very much. How we can do this is not easy to have these benefits, ecological, social, economic benefits. Okay, so agroforest systems can make all these things. Control nitrogen fixation, water infiltration, pollination, biodiverse hold, CO2 sequestration, production, weed control, soil properties, fruit production, nutrient cycling, every these things we can modify by inserting something that can last over decades, like trees. Okay, so as we talk, you see that trees have different structure, and this old work from Larcher is from Austria. You can see that the Pinus has an ability to take all this sun. The light interception is very much. Besides the olive trees in the right lawn, it doesn't need so much sunlight, okay? So the interception is much poor. And on the other hand, you have single structure and the community structure. As you can see, sunflowers, corn, pines, and the boreal forest here. You can see how much lost energy in the soil in here in sunflower. 19% reached the soil, and corn 7%, and in forest only 2%. So how can we reach this efficiency? By making a new structure, okay? So let's see how we understand plantations today. We are using only 2 dimensions for planning, okay? Rows and between rows. So we are dealing with the space in a flat sense. This is what I'm trying to change the mind, no? So in Brazil, we used to plant big plantations here, 3 by 2, and you can see all this thin layer of the canopy. It's very shallow. The trees are very thin, maybe 15 centimeters here, okay? So all this is for production of pulp, cellulose pulp. So in nature, we have this situation, huh? Colonization, planting, growth, harvest, then you come back to the disturbance. This is very bad for ecological process. The disturbance in the bare soil is rare in nature, but we are making this a lot of bare soils, a lot of destruction. So you don't have live plants, living organisms to respond to the environment, okay? So if you think about that and all these problems, and if you try to make something different, huh? So one day I was in the street and I say, stop the car to my wife. And I saw all these leaf walls so different and this edge effect. You see how much more leaves you can have. And one time I was in the experimental station, people didn't care about the border. They say they measure only the thin plants. I said, these people is not very clever because the biggest trees were in the edge. So my problem was to obtain bigger plants and not thinner plants. So I invented, based on these microvilles on our intestine, how we can increase the surface in order to absorb, in this case, nutrients, and in our case, to absorb light, energy. And you see here, this photograph, you improve greatly the interface between intestine, nutrients that you eat, and yourself. Okay? So based on that, we could increase light absorption by making something that could capture the energy, the movement, okay? By developing a new structure. So we made an experiment. Here is our research station. We made this quite near, 2 hours from here. The WAVE project. The WAVE project is trying, huh? It's still growing up. 3-dimensional structure and try to make this new function. We use 4 dimensions, width, length, height, and time. You don't see many times people using time as a project thinking. And they never go to square zero, to this disturbance, and they maintain the ecological project, okay? So what have we done? We have this space here. I will show you a cross section. So you are planting in the border in different times, okay? 1A, 1B, then 2A, and then using between the rows with other things. The dark one is the eucalyptus trees. So you are going to closing each year. You are planting inside the rows and harvesting the inside, having money, and letting the older trees growing more. you don't need to be only eucalyptus. You can make this for any kind of plants. So here the years we have planted, okay, up to 7. The 70, the youngest in the middle. The idea that when these bigger trees are harvested, then you have a gap, like in forest gaps, okay? So we are trying to simulate also ecological process. Taller trees, then you make a gap, the other trees respond, and you can plant many things inside, okay? So you can see here, in 1915, how much light get inside of this structure. You see the next one. You have many grasses, okay? You can see the environment, how it's changing, and you are going closer. So the difficulty of this is to know how wide is this wave length, okay? And how much this can be high. So we are going to show some research from this beautiful lady, my former student on Master Science. And we see Gabriela results here. So what we are going to do? Here is the monocrop. We call this middle one short wave, and this is the long wave. The wavelength of the distance from between the highest trees, okay? So you can use mathematical model. I will share this with you can see. You can predict how much light get inside of these edge rows. Okay. Gabriela made many dendrometric tapes to measure the growth of these trees. You can see here, this a little bit older, 2017, with trees with only 6 years. You see how big these trees are here, okay? It's maybe 20 something in that time. We are now obtaining 32, 35 centimeter diameter with 10 years. So what you can see here, the deviation, monocrop and long wave, you see at the same age, it was year 1 and year 2, okay? The first rows and the second rows planted inside. In monocrop, it was about 15 centimeters DBH, and long wave, 20, okay? It's very big. Here, 22 canop volume, cubic meters. Besides, long wave trees had 90. Almost 5 times bigger. And the year 2, DBH was about the same, all this condition. This means that the tree in the second year is a little bit too much shade. You need to understand this in order to know where to put. And you see the canopy of the monocrop is a little bit bigger here, surface canopy and volume canopy. So how this trees grow along 2 years, we measure this by these dendrometric tapes. Here the blue lines are rains. You can see the year 1 plants, how bigger response can be, while monocrop plants are very low. Okay, it's far away, growing much bigger tax. And year 2, that one, the second, it's a little bit shade. You see, monocrop is a little bit better. Okay, monocrop is this square one. Yeah, more or less together, sometimes bigger. Okay. So what we can be practical in this? Monocrop, short wave, and long wave. Short wave, we have 4 lines for each side. Long wave, we had 7 lines of trees. So while we have a monocrop with in 2016 with 100 kilos dry weight for each plant, in long wave, 240. So it's an improvement of 2.5 times bigger, 250% bigger trees. Bigger trees means better wood, better revenue, okay? And you see here, monocrop is the square one. With the time, it's saturated, okay? You see the saturation point, the trees are not growing, it's slow growing because all the area is occupied, it's not going to have a better growth. While plants in the wave are growing and growing, growing, and didn't stop growing. You see how the potential to grow this, okay? Nowadays, what we are doing is a simulation process using architecture models to simulate light interception like that. You can see this program now is Rhinoceros, okay? And you can see how the light is intercepted, okay? This is very good for bigger latitude places because you change the angle of the light interception. So if you are like where you are, Uttar Pradesh, in Pradesh, near Delhi, you see that the sun is always in the south. So you can plan how to intercept this and change this angle to increase the light interception. So, conclusions. We can generate more products, more income for the people. Improve solar radiation interception use, change the interception plane, as I told you. Increase land use efficiency, intensification. This system can be used for many species. I know India has facing some problems with the eucalyptus, so you can use another species, okay? Don't be tired with this. Increase the productivity, maybe even better for high latitudes, I told you. Arouse people interest, can be adapted to many systems, diverse species. People can be integrated and be allied to better recover this ecological process, and we need to leave static system for dynamic systems, okay? So we need to stop thinking 2 dimensions, flat, only rows and inter-rows, and need to think in 4 dimensions. Length, width, height, and time. Okay? And to finalize, this is a reinterpretation of the drawing, this first drawing of a wave. We need to reinterpret it. This is by a child. And this is the end. I would like to thank you very much, Ahmed University, for this, Madhya Pradesh. Thank you very much.
Thank you, sir, for such an informative and enriching session. We are extremely honored by your benign presence. We are privileged to invite a distinguished scientist and academician, Professor Dr. M.P. Kaushik, Co-Vice Chancellor Research, Amity University, Madhya Pradesh. With a remarkable career at Defense Research and Development Establishment, Government of India, where he retired as director and outstanding scientist. Dr. Kaushik has represented India globally and has been honored with prestigious awards like DRDO Scientist of the Year and the Agni Award for Self-Reliance. So we now request you to kindly share your concluding remarks with us.
Thank you very much for the kind introduction. And good afternoon to all of the members, those who are attending online, particularly Dr. Maria, as well as Professor Sairo Rigi. Welcome to the Amity University platform, and then thank you so much for sparing your valuable time and coming over here and talking to us on various aspects of the sustainable technologies which are existing. Now, in the last 2 days, inaugural session as well as the valedictory session, large number of papers have been presented, and various aspects has been covered, from bioremediation to that of the culture, the microbial degradations, whatever Madam Maria just mentioned over here, and then how the plantation can help in terms of the economy, which the Professor Siro was mentioning. No doubt we are looking for different kind of varieties in terms of plants as well as trees, which should use minimum amount of water. There was issues in Indian scenario where eucalyptus plant was in using more amount of water from earth water, and that's why the water level was going down and it was not considered suitable. So as you rightly mentioned that we have to look for the different varieties or varieties which use minimum amount of groundwater and can look for, and then the type of the leaves wall which you have showed and mathematical modeling which you mentioned is really very impressive and very knowledgeable, and then awareness has been created by you. So thank you very much. So I'll talk two different aspects of which I have learned over the last 2 days. One is the burning topic which is required by for VIKSIT Bharat, that is developed India. When we talk about VIKSIT Bharat, we are talking about developed India. What are the sustainable technologies we have to emphasize and we have to work on it? Whatever comes to my mind, I have worked a little bit on these technologies, but somehow that it could not come to that level which we should have gone for commercialization, but there's an empty scope for taking up. That is microbial desalination of seawater. Yesterday when I was talking in one of the program, yesterday itself, when I was concluding I was giving the concluding remarks about the 2 talks which the experts were giving, I mentioned that water is an important commodity for life, for survival, particularly potable water, drinking water. And if at all the Third World War is going to be fought, it is going to be fought in water. Water is going to be the crisis in the days to come. I'm talking about potable water only, not any other kind of water. And the second important aspect of is energy, with Professor Zero was just mentioning how energy can help us in protecting our life and how energy can be used or can be produced with the help of biomass, which he was telling with the different kind of trees where the leaves or you take the size of the plant, which can convert into large amount of biomass. So the microbial desalination of water is an important aspect of it, which we have worked at. It means if we can make seawater into potable water, which with the microbial cell, and ultimately convert the seawater into potable water, that is a tremendous technology which we can work on it, and it has been done. We have used different kind of seawater and microbial, which are salt-loving bacteria, which normally takes the salt, consumes salt, and then on the electrolysis, the anode as well as cathode, the sodium and chloride ions are being separated, and the amount of energy is being generated. It is good enough to generate sufficient amount of light, at least for the in-house use, it can be generated. So this is one of the area where people need to work on it, and then particularly in developed India, this type of a project should be taken, and it is encouraged by the government of India with the help of Niti Aayog. Large amount of funding is also available in this. Another is the bioremediation or biodegradation, and what you said, the bioenergy which is being produced by the biomass, or biomass can be of any kind. No doubt it's whatever just mentioned by the Professor Zero as well as Madam Maria. Microalgae is also one of the source, and at the same time, if you take the agriculture waste, if you take the animal waste, if you take the sewage water, if at all put together, and you can make of a particular size, that particular particle size is being done, and that is being fermented for over a period of time, along with that bacterial microbial growth, then amount of methane which is being generated is good enough to convert into the energy, that is the electricity. And when electricity is being produced, there is a model example or model pilot plant has already been established in one of the town in Haryana, that is called Sirsa, where at least a 5-kilometer area of the population is being given the electricity with the help of methane, which is being produced by the microbial degradation. And the amount of water which comes out of the microbial degradation is being used by the farmers, and it is being used as a fertilizer, and the percentage yield has also gone up at least twofold from whatever was used by the normal water, normal irrigation water, or the canal water. So you can understand that it means if we take all that waste also, it means we are taking care of the environmental also, as well as at the same time we are producing energy. So that will go a long way. So these are the 2 sources are there which we can take up this type of project, particularly even for Gwalior people also. We suggested to Gwalior Nagar Nigam that we can take the complete the agriculture waste, human waste, as well as the animal waste, which is normally can be converted into biomass, and ultimately electricity can be generated. So these are the important aspect of it. And microalgae, which Professor Mario was mentioning, is one of the area where the large amount of biomass is being produced during, particularly in the seashore area, where we have seen some of the microalgae which are of of practical importance or which can be used as food products, but at the same time, it can also be used for the energy productions. So these are the areas. And now I'd like to compliment the organizers, particularly Dr. Dwivedi, Dr. Rai, and the other faculty members of the Amity Institute of Life Sciences, particularly environmental sciences, as well as biotechnology and other departments who have contributed very significantly towards the growth, and all the speakers who have come from all the way and talked on the different aspect of the environmental sustainable technologies, which is required for. There are so many other technologies which are there, which still can be taken up, but these are the two important areas. The water and the energy is the areas where we should work on it. So I am very thankful to you for all the delegates, those who have come all the way, for all the students. And I am sure this international conference on the sustainable development of sustainable technologies would have given new ideas to the students, to the research scholars, to the faculty members, and it will help them in carrying out some of the new innovations and inventions in the days to come, particularly for relevant technologies. So thank you very much once again, and Jai Hind.
Thank you, sir, for your concluding remarks and valuable insights. Ladies and gentlemen, now we come to the much awaited moment, the announcement of Best Poster and Best Oral Paper Presentation. First, we'll be announcing the results for the poster presentation. The third position is secured by Adit Singh Bagga from Guru Nanak Dev Engineering College, Ludhiana, Punjab. Congratulations. The second position is backed by Shikha Diman from Amity Institute of Biotechnology, Amity University, Gurugram, Haryana. Congratulations. And the first position for the poster presentation is backed by Anu Kumari from Maharishi Dayanand University, Rohtak, Haryana. Once again, congratulations to all the winners for the poster presentation. Following this, now the results for the oral paper presentation are as follows. Third position is backed by M. Aarti from Sri Ramchand Institute of Higher Education and Research, deemed to be University, Chennai, Tamil Nadu. Congratulations. The second position has been backed by Anup Sharma, Farm Machinery and Post-Harvest Technology Division, ICR, IGFRI, Jhansi. Congratulations. And the first position is backed by Madhurima Jodhar, SRM Institute of Science and Technology, Ramapuram, Chennai. Congratulations. So once again, congratulations to all the winners. To formally conclude the session, I invite Professor Dr. Kuldeep Dwivedi, convener of the conference and head of institution EVS, Amity University, Madhya Pradesh, to purport the word of thanks. Sir, please.
Good afternoon to everyone. It's my pleasure and an honor to propose the vote of thanks at the end of this 2 days international conference on sustainable technologies for VIKSIT Bharat. And at the onset, I would like to congratulate the winners of the poster and the oral presentation, because the conference is organized for the participants. We, the organizers, as well as the invited speakers, are there to facilitate and to bring to them the recent trends and technologies which are to be investigated by them. So even the array of papers that were presented reflected on the main themes of the topic, and the themes of the papers which have won the prizes are also in concurrence with the themes of the conference. So I congratulate them once again. And on behalf of the Amity University Madhya Pradesh, as well as the organizing committee of this conference, I extend my sincere gratitude to the chief guest of the inaugural session, Sri Sangapri, Municipal Commissioner Nagar Nigam, Gwalior, guest of honor, keynote speaker, Padam Shri Awardee, Professor Dr. Arvind Kumar, former Vice Chancellor, Rani Lakshmi Bai, Central Agriculture University, for their valuable presence and address during the inaugural session and for setting the tone of the conference for the 2 days to come. I would like to express my sincere thanks to Dr. D.K. Shukla, Scientist F, Department of Atomic Energy, Devi Ahilya Viswavidyalaya Campus, Indore. Dr. S. Ganpati, Professor, Department of Environmental Law and Management, Anna University, Chennai. Dr. P.K. Pathak, Principal Scientist and Head Division of Farm Machinery and Post-Harvest Technology, Indian Grassland Fodder Research Institute, Jhansi. Dr. Anish Pandey, Associate Professor, Government Model Science College, Gwalior, for sparing their valuable time and delivering plenary lecture in the technical session, which was organized in an offline mode. I would like to express my sincere thanks to Vinay Kumar Tyagi, Scientist D, National Institute of Hydrology, Roorkee, Dr. P.C. Avilas, Associate Professor, Institute of Environmental Sciences and Sustainable Development, Banaras Hindu University, Varanasi, Professor Dr. Nagaraju Madela, Department of Biological Sciences, Porto Vigo, Ecuador, and Professor Suresh Kumar, Department of Botany, Ramjas College, University of Delhi, Dr. Rakesh Bhutiani, Associate Professor, Department of Zoology and Environmental Sciences, Gurukul Kangri Viswavidyalaya, Uttarakhand, Dr. Hemant Nothalapati, Associate Professor, Department of Life Sciences, Faculty of Life Sciences and Environmental Sciences, Shimen University, Japan. Dr. Asutosh Tripathi, Professor and HOD, Department of Environmental Sciences, Nagaland University, Lumani. And Dr. Santosh Bhukal, Department of Environmental Sciences and Engineering, who took valuable time from their busy schedule and joined us online in the online sessions and delivered the plenary lectures. My sincere thanks to the guest of honor of the validity session, Professor Dr. Luciana Maria Cola. Heartfelt thanks to Professor Dr. Ciro Abdurrigi from Brazil for the gracious presence in the validity session and for delivering a very insightful lectures, both of them, particularly on the very important aspects as far as sustainability in the present times, particularly in the era of this climate change is concerned. Microalgae as the future food and for the generations, and Dr. Ciro for suggesting a agroforestry systems consisting of different species having the ability to absorb the sunlight spectrum and sustain the growth under diverse agroecological zones. And the selection of species, particularly differing in the different geographical areas, will definitely give a very good model suggested by Ciro as far as sustainable agriculture is concerned, because agroforestry is a medium through which we can obtain evergreen revolution, although we have passed through the green revolution and attained sustainability, self-sufficiency in food grain production, but the need for new avenues of fodder, fuel, and conserving our ecosystem, agroforestry is the answer to it. So I congratulate Ciro for delivering such an insightful lecture to us, and the participants must have benefited by his lecture. I would like to put on record my sincere gratitude to the Honorable Pro-Chancellor, Lieutenant General V.K. Sharma, for his able guidance and mentorship. My sincere thanks and gratitude to the Honorable Vice-Chancellor, Professor R.S. Tomar, for his unwavering support and insights, which have significantly contributed to the success of this event. I'm also grateful to the Honorable Pro-Vice-Chancellor Research, Professor Dr. M.P. Kaushik, for his valuable advice, suggestions, and encouragement. He has been kind enough to grace the validity session and deliver the concluding remark and summarize the outcomes, at least, of the deliberations that have taken place in the 2 days, which reflect his keen observation and insight, and we will continue to look towards you for guidance and further improvement in the coming events. I'm thankful to all the academicians, researchers, scholars, and students for their overwhelming support and participation in this conference, which has culminated in the successful organization of this event. I'm thankful to the ID team, which has provided us support as far as the online session conduction is concerned. I am grateful to my team members, Professor Dr. Swapnil Rai, the co-convener of this conference, Dr. Ritabrata Malik, Dr. Nidhi Shukla, and Dr. Abhishek Bhardwaj for their tireless work in organizing this conference. My sincere thanks to Sonal Singh, Mangalam Soni, research scholars of our department, who have been working in the front as well as behind the scenes to make this conference a success. Special thanks to Ms. Anvesha Singh for the flawless conduct of the inaugural technical and validatory sessions. And last, I thank each and every one who are present over here, who might not be present over here, who have helped us in one way or the other in the successful conclusion of this event. Thank you. Thank you, each and every one of you. Jai Hind, Jai Bharat.
Participants can choose the raise hand option if they would like to speak.
Yeah, any participant would like to have some feedback about this conference, he's welcome. He or she may raise his or her hand and give the feedback of this conference. Anyone? You can just raise your hand. Okay, there's Anu Kumari. Please unmute her and open a camera so that she can be visible to us. Right.
Am I audible?
Yeah, you are.
First of all, a very good afternoon to everyone present here. I am Anu, research scholar in the Department of Environmental Science from Maharishi Dayanand University, Rohtak. I am deeply honored and grateful to have received the Best Poster Award at this conference organized by the Amity University on Sustainable Technology for the VIKSIT Bharat. The platform has provided me with a valuable opportunity to represent my research and engage with the researcher and fellow participants from around the world. The session was insightful, the discussion was intellectually stimulating, and overall the organization of the event was commendable. I sincerely thank the organizing committee, session chair and judges for recognizing my work. Thank you once again for this incredible honor. Thank you so much.
Thank you, madam. Thank you. Yeah, there is Dr. Madhurima. Yes, please kindly unmute her and.
A very good afternoon to all the delegates, to all the organizing committee of this international conference. Thank you everyone for giving me a chance, especially Ritobrato sir, because without him, I wouldn't have been able to participate in this conference today. And thank you for giving me the first position in the oral presentation. I'm really honored. I'll be doing much more better for the society in the near future. And I would thank for all the senior delegates who will have presented from outside India International. They are very nice as well as they are having very much knowledge, which has helped me enriched my knowledge also. Thank you, everyone. Thank you once again.
Thank you, Madhurima. Thank you. Anyone else?
No.
Okay then. Thanks once again, Madam Luciana and Dr. Siro for your presence. And we are really thankful for your presence and you having delivered what you are actually working in your labs. And I definitely hope that the young students and scholars would have immensely benefited from the deliberations of both of you. Thank you. Thank you very much.
Very nice, Dr. Kudpil and Dr. Ray. It was very great to be here. Next time, we need to go to India. I have been twice.
Yeah, definitely, sir.
Karnataka and New Delhi.
And I need to tell you, Dr. Siro, just 100 kilometers from Gwalior, where we are located, there are two very important institutes related to the work that you are doing: Indian Grassland and Fodder Research Institute and Central Agroforestry Research Institute. And I am a student of that one of those institutes. So I feel very affiliated to you and the type of work that you are doing.
Nice. Next time I will show you something about chicken agroforestry.
Okay.
Nobody's doing that.
That's great.
Definitely.
So many things that we can do, huh?
Yeah. Okay. Thank you, madam. Thank you, sir. Thank you.
Thanks for the opportunity.
Okay, thank you.
Thank you, Professor.
Sir, shall we close the meeting?
Yeah, we can close.
So, Arvind ji, please close the meeting. Thank you so much. Thank you.
Thank you. Thank you all.
Bye.
Thank you.
Thank you, everyone.
Thank you.